A very big hello to all my wonderful PU2 champions. I hope every one of you is doing absolutely great. So I see lots of comments around biology this time, and I'm quite surprised with that. Technically speaking, I am happy as well. But please keep all your worries away, or you can probably give it to me, and be rest assured that you are going to score great marks, my dear children. Why to worry when JJ is here, right? And in this video, as I said, we'll talk about the 1-marker questions, because MCQs is the place where most of us lose our marks. And yes, I'll try my best. I just have one request for you guys, that is, please tolerate me for the next few hours. And if at all you are ready to tolerate me, probably you might be able to do really good with the MCQ part of the question paper. Yeah, and one more note that I want to put over here, disclaimer, and that is, it's not like the exact question is going to show up. Probably some of them might show up, some of the concepts might be repeated, because all these questions have been taken from the model question paper, from the question bank questions, from the previous year question paper. So I tried my best to cover all the nook and corner, but your NCERT is very, very vast. If at all I go around talking about each and MCQ of each and every line of NCERT, probably YouTube would not allow us to upload any videos, because it would be that long. So my dear children, keeping all this thing aside, whenever I show you a question, I just want one thing from you, and that is pause the video and try to solve the question by yourself. And if at all you're liking this lecture, do not forget to click on the like button as well. Yeah, and share it with your friends as well. So let's go ahead with the first chapter that we have, that is sexual reproduction in flowering plants. And here goes the first question. Statement number 1. Pollination does not guarantee the transfer of right type of pollen. Pollination means what? Transfer of pollen grains from the anther to the stigma. Now, it can be right type, it can be wrong types coming from some other flower and things like that, right? Come from other species also altogether. So, pollination does not guarantee the transfer of right type of pollen. Yes, it does not. This is actually a true statement, right? So, this is true statement. Next, compatible pollen pistil interaction guarantee double fertilization. Listen to me. So, basically, onto the stigma, the pollen grain is going to come and land, right? Now, they form the pollen tube. And the pollen tube will be coming over here into the ovary. Inside that you have the ovule. Inside that you have what? The female gamete. That is the whole story. Pollen tube will be having the male gametes as well, right? When the male gamete comes and fuses with the female gamete, you say that the syngamy has taken place. Zygote is being formed. Yes, that is the first fertilization. Now, the next fertilization happens when the next male gamete comes and fuses with the polar nuclei. That is known as triple fusion, because 3 nuclei are being fusing up there, right? When 2 times fertilization happens, once with the female gamete, another one with the polar nuclei, my dear children, we say that double fertilization has taken place. And here the statement is, whenever there is a compatible pollen pistil interaction, okay, it will definitely guarantee double fertilization. Do you think so? What do you think so? Suppose it's not just one pollen grain, but multiple pollen grains landing. They are all forming tubes, my dear children. So does that mean that all of them are going to do double fertilization? No, Baba. There is no guarantee that compatible pollen pistil interaction will definitely make sure the double fertilization will occur. Sometimes the pollen tube form, but they stop right there. Other the pollen tube can form, go ahead. So we never know, that can happen, that may not happen. So there is no guarantee to it. So this particular statement is wrong, because it says that the compatible pollen pistil interaction guarantees double fertilization. The answer is wrong. So statement 1 is correct, but what about statement 2, Baba? Statement 2 is going to be wrong. Understood? Read the question carefully. Okay, and then everything is going to be sorted. Yes. The next question that I have over here is statement number 1 says, apomixis is the production of seeds from unfertilized ovules. Is it? What do you mean by apomixis? In apomixis, what is going to happen, my dear children? Suppose this is the ovule. Inside the ovule, what do you have? You have the embryo sac, right? Inside the embryo sac, what do you have? You have the female gamete. You have the female gamete, you have 2 antibodal cells and blah blah blah, so lots of things like that. Usually the female gamete which is there, it is haploid in nature. It has half the number of chromosomes, and it actually waits for the male gamete to come, so that both the haploid cell comes together to form a diploid zygote. Diploid zygote. That is how it usually goes. But you know what? There is a change in the trend. Over here, this particular seed thinks that okay, or basically this particular plant thinks that okay, I do not need any male partner to form this zygote. Why not? Why not? Here, instead of being haploid, let me be just diploid. Let me be just diploid. So the egg cell which is there, the female gamete which is there, my dear children, what is going to happen? It is already diploid in nature. So does it need anyone else to become a diploid zygote? No, Baba. It itself transforms into what? A zygote. A zygote, and when a zygote is formed inside the ovule, you will now not call this as ovule, rather you have a different name for it, which is known as a seed. Whenever a zygote is formed inside the ovule, you will now call this as a seed. Over here, even without the help of the male gamete, it formed the zygote, and now it is going to be known as a seed also. So apomixis is the production of seeds from unfertilized ovules. Do you think that this particular ovule underwent fertilization? No, there was no fertilization. So this is correct. Is the production of seeds from unfertilized ovules. This particular statement is absolutely correct. Now next thing, embryos produced from the apomictic seeds are not generally identical to the parent plant. What? Listen to me carefully. Here only a female parent is being involved. Female gamete is being involved, right? There is no fusion of gametes. If there was fusion of gametes, I'll say it's sexual reproduction that is happening. And at the end products of sexual reproduction, you always get someone new who is having a combination of genetic material from both the parents, right? At that particular point of time, I'll say that they are not generally identical to the parent. For example, I'm not exactly like my mother or my father. Like it's a hybrid, right? Why? Because sexual reproduction is being followed in the case of human beings. Now here also, do you think sexual reproduction is happening? There is no fertilization, Baba. So there is no sexual reproduction. If there is no sexual reproduction, can I say it's a type of asexual reproduction? Yes. And in asexual reproduction, what happens? Only one parent is involved, and the progenies which are going to be formed, they are going to be just like that parent only, because they are just passing the genetic material, right? So they are going to basically form clones, and I'll say that they're genetically very much identical. So embryos produced from apomictic seeds are not generally identical to the parent plant. Wrong statement. Again, a wrong statement. Statement number 1 is correct, and statement number 2 is what? False. Got it? Yes? Yeah. Now, the moment you feel that, okay, ma'am, speed is going a little bit fast, what you can do is go to the settings. Okay, I do not know on what side it is. Go to settings and make my speed less. The probable reason that I might be going fast is that there are so many questions to be completed, and probably I'm thinking in the back of my head that you do not have much time to watch this video. So that could be the only reason. It's not because I need to do it fast. I am usually someone who goes really slow while teaching. Okay, so it might be just because of for you guys. So please forgive me if at all it is happening, and please change the settings out there. Yeah. Okay. Now, even in the absence of pollinating agents, seed setting is assured in. Seed means fertilization has taken place probably, right? That means zygote has to be formed. That is what you mean by seed formation. Now, zygote to be formed, obviously you need the pollen grains to come and land on the stigma such that it can give the male gametes, and the male gametes and the female gametes can fuse. But who will bring the pollen grains? Obviously, it has to be some pollinating agents. But here it is said that even in the absence of pollinating agents, seed setting is assured. Seed definitely is going to be formed in which plant? Cucurbita, papaya, commelina, maize. In which condition do you think this is definitely going to happen? Without pollinating agents, seed setting. My dear children, if suppose the flower is like this, closed, it has the female part, it has the male part. From the male part, the pollen grains lands on the stigma, that is on the female part. Here, do you need any pollinating agents? No. That means these flowers has to be what? Closed. Ma'am, are these flowers for real? Yes. What do you call them? You call them as clistogamous flowers. Okay, you call them clistogamous flowers. Here Cl stands for what? Closed. Here Cl is going to stand for closed. Okay, so definitely pollination is going to happen, and after that seed setting is also going to be happening. Yes, the only drawback that is going to be there is like it will be just like the parent plant. So parent plant, suppose it has some toxic character, it cannot be pest resistant or this and that, any bad characters are there, it will be just passed on. Okay, and in your NCERT there are 3 plant's name given, and you have to remember it. I remember it like this: cow, but with a V. So C for Commelina, O for Oxalis, and V for Viola. Commelina, Oxalis, Viola, they have two types of flowers. One is the closed flower that you just saw, and one is the open flowers which are there. And here the normal type of pollination will be happening. They will require pollinating agent. They sometimes not require pollinating agents. Okay, so it all depends. Anyway, it is going to be open. This type of open flowers are known as chasmogamous flowers. What do you call them? Chasmogamous flowers, and they are known as clistogamous flowers. They are usually found towards the root region. This is given in your NCERT. Low key, sometimes you might not see it. So please understand, they are mostly found towards the root. Okay, probably within the soil itself they might be found, or just above the soil they might be found. Clistogamous flowers. Okay, so over here you have the answer, and that is Commelina. Commelina is the answer for this. And as I said, here I have put in a real-time picture. These are the open flowers that are known as chasmogamous flowers, and here you have the closed flowers. If at all you see, it's just above the the soil, yeah, or it can be beneath that also. Fine. Yes. Next question. Select the correct sequence of anther wall-lace. Anther, if at all you have seen, my dear children, there are multiple wall-lace, 4 of them are there. Okay, I'll require you to remember it like this. It's very easy. So have you been to ear, nose, throat doctor whenever you have any problem in the ear or nose or throat or anything like that? You go to an ENT. You go to an ENT. Similarly, over here, you have EMT. Okay, so E for epidermis, that is going to be the first layer. So let's see. Epidermis, epidermis, epidermis. So this one is the odd one out. Okay. Next one. After epidermis, you have endothecium. Endothecium, endothecium, endothecium. Only this one has. Let's go ahead. After endothecium, you have middle layer. Okay. And after middle layer, you have tapetum. Oh my God. So your answer for this particular question is going to be B. Always remember, epidermis, endothecium, middle layer. These 3 of them are best of best friends. Why? Because when the anther, which has pollen grains, my dear children, the anther has to dehisce, basically break open, so that all the pollen grains go outside. Who are the 3 members of this particular gang who break open for the anther? It is basically epidermis, endothecium, and middle layer. So I'll say that they are responsible for dehiscence of anther, or for the breakdown of anther. What about tapetum, my dear children? Tapetum, on the other hand, is Papa guy, is there for nourishment. Nourishment of what? The sporogenous tissue. So it's a nummose layer, right? So here you have the sporogenous tissue, which is made up of multiple cells, microspore mother cells, which will later form the microspores, which will later form the pollen grains. So to provide the nourishment to that, you have tapetum. There are certain features of tapetum that you need to remember. First of all, since it has to provide nourishment, it will be having a very dense cytoplasm. Okay. Apart from that, what about the nucleus? Nucleus, suppose it's a cell, they have 2 nucleus. That means it's a binucleate condition. What condition? Binucleate condition. 2 nucleus is found over here. So that means karyokinesis took place. Nuclear division is known as karyokinesis. That took place. But cytokinesis didn't take place. Cyto means cell. Cell division didn't take place. Okay. The karyokinesis took place out there. So dense cytoplasm is there. They have binucleate condition. This is a feature of tapetal cells, which is there for nourishment of the sporogenous tissue. Okay. Or the cells of the sporogenous tissue. Works? Works, ma'am. Awesome. Okay. Now, so I have spoken about this already, and I have given you, this is a question bank question. Number of the question bank question is also given down there. Now, select the correct sequence of events in microsporogenesis. In microsporogenesis, how is the thing going on? So let me, please try it out by yourself. Pause the video. I'm going to go ahead. You might see the answer, but I just want to check if at all an animation is given there. Yes, it is there. So I'll explain you with this animation. That is much easier. So my dear children, as I told you, in sporogenous tissue, you have microspore mother cell. Microspore mother cell. They are what? They are diploid in nature. They are having diploid number of chromosome, right? They undergo meiosis. Meiosis happens in 2 stages. First meiosis, second meiosis. When the first meiosis happens, the number of chromosome, it becomes half. That is why you call meiosis as reductional division. Okay. Now the next meiosis happens. There are 2 cycles that happens. First meiosis, second meiosis. Again, do you think it will get half? No, Baba. It will remain the same. N. only thing that you need to remember is that these microspores which are there, they are together, they form a tetrad. Tetrad means 4 of them are together over here. They are microspore tetrad. That is how it is being formed. Later on, when the anther is going to get dehydrated, when the water is going to be lost, all of them get separated. And after getting separated, what do they form, Baba? Each of them individually are going to form our own pollen grains. Our own pollen grain. That is the story. So sporogenous tissue first, then microspore mother cell, then microspore tetrad, and finally you got the microspores that will later form the pollen grains. When these type of question comes, read all the options carefully. All the options carefully. So they might miss this thing, but write all this thing in sequence, right? So please check out for that. If at all this is being missed and everything else is correct, it's okay. It's okay. If at all, this is common for all. Sporogenous tissue has never been mentioned over here. You should know what is the next thing. You get it? That is what I am trying to tell you. Yeah? Yes. Okay. Now the next question. From the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. What is a female gametophyte? The pollen grains that we saw, my dear children, do you think that was a male gamete? No. It forms the male gamete, which is why it is known as male gametophyte. Similarly, in the case of females, you have a female gametophyte which is going to form the female gamete. This female gametophyte has a name. It is known as what? Embryo sac. It is known as embryo sac, and it looks something like this. So embryo sac also, let's read the options first and then I'll go ahead with the answer, because I think again animation is given over here. So it is 8 nucleate and 7 celled at maturity. 7 cells are there but 8 nucleus, how can that be possible? Do you think that is possible? Okay, let's see. It is free nuclear during development, as in the nucleus. Whenever a cell is here, so many cells are being formed, right? 7 cells are being formed. It says that the nucleus of these cells first divided and then later on the cell walls formed around it. That is what you mean by free nuclear division. So it's not like immediately cell wall was being formed. First of all, the nucleus divided, later on the cell wall was being formed. That is what you mean by that. Okay, free nuclear division. Now, it is situated inside the integument but outside the nucleus. What do you think? It is situated inside the integument but outside the nucleus? According to what I know, it goes like this. So, you have the ovule. The outermost thing that you have is the integument. Inside that, you have the cells of the nucleus. And inside that, you have the embryo sac being formed. So this is the embryo sac. Okay, there is a female gametophyte. Outside that you have nucleus. Outside that you have integuments. But what is given over here? It is situated inside the integuments, sure, but outside the nucleus. Embryo sac is not outside. Embryo sac is inside the nucleus also. So this is also correct. It has an egg apparatus situated at the chalazal end. What is egg apparatus? Egg, my dear children, the female gametophyte will be having a female gamete, right? The female gamete is guarded by 2 cells which are called as, so what do you call them as? I forgot the name. Oh my god. Synergids. Yeah, synergids. So synergids plus the egg cell, that is the female egg, you call this as what? The egg apparatus. And egg apparatus can never be located at the chalazal end. Mostly, the most important things are located towards the micropylar end. So always remember, the integuments have an opening over here which is known as micropyle. Okay, and the opposite side of this is known as chalazal end. Everything important is going to happen up over here. Your egg apparatus is going to be forming up over here on the micropylar end, not on the chalazal end. So this is definitely going to be wrong. But what about these 2 options? We have to see that, no? Is it 8 nucleate and 7 celled at maturity? Let's see that. Okay, so over here, understand, so just how you have microsporogenesis happening in the case of males, similarly you have megasporogenesis happening in the case of females. At the end you get 4 megaspores, okay? 3 of them are going to degenerate, only 1 is going to be left, and when the one which is there, it is going to go ahead and form the embryo sac. You call this development as monosporic development. Why? Because only 1 guy is involved over here. Monosporic development, right? So this is the cell. This is the megaspore, the only megaspore that is left. Here you have the nucleus of that. The nucleus undergoes division. How many times? Thrice. So first division, you got 2 nucleus. Second division, again you got 4 nucleus. Third division, 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus are going to be formed. At the end of this, you get 8 nucleus, my dear children. Very, very, very important thing. And over here, you do not see any cell wall formation, which is why I'll say it is what? It is free nuclear condition. It is free nuclear condition. That means there is no cell wall formation around it immediately. And 3, mitotic division is what you say. 3, mitotic division. Now, later on the cell wall and everything is going to be formed and you will see something like this. So basically, 3 of the cells go up there at the chalazal end. This end that you see is known as the chalazal end. And these 3 cells with 3 nucleus, you call them antipodal cells. Similarly, 2 cells come over here, you call them synergid cells, each of them having their nuclei. Okay, so 3 plus 2, 5 cells are done. One more cell comes in and that is the egg cell. Now, that also have nucleus. Yeah, so 5 plus 1, 6 cell. Now, where is the 7th cell, my dear children? Where is the 7th cell? This whole thing that you see over here is the 7th cell. So you have 7 cells, but this 7th cell has 2 nucleus. Instead of having 1, we have 2 nucleus. So all of them individually had 1. Until 6 it was fine. Until 6 of them it was fine. Now in the 7th cell there are 2 nucleus. So this is going to be the 7th nucleus and this is going to be the 8th nucleus out there. You call them as polar nuclei. 2 polar nuclei of the central cell. So isn't it 8 nucleus and 7 cell end condition? The answer is yes. Okay, the answer is yes. This is very, very important. So all the statements eventually are going to be correct. So let's see. So A and B. Yes, it is correct. A and B is correct. Okay, and what about so did I actually say it is correct? Oh my god. I explained you something else, and if at all I have said that this is correct, I'm so sorry, okay? So A and B are the only one which is correct because it is situated inside the integument but outside the nucleus. No, that is not true because it is inside the nucleus also, yeah? And it has egg apparatus situated at the chalazal end. Baba, this is the egg apparatus that you see, okay? Oh, just a second. Yeah, this is the egg apparatus that you see. It is towards the micropylar end, not towards the chalazal end. As I told you, everything important happens towards the micropylar end. Even the division starts in the micropylar end stage only. Okay, now let's go ahead. And over here, we had the 7th question. Identify the correct sequence of arrangement of cells in mature embryo sac micropylar end. Micropylar end. So in micropylar end, what do you have? Synergids is the first thing that you have. Then you have the egg cell. Then you have the very big central cell. Then after that, at the top, what do you have? You have the antipodal cells. So this ends the answer. Simple. Antipodal cells cannot be in the micropylar end, wrong. Synergids, then central cell, then egg cell, no. Synergids and egg cell comes together, you call this as egg apparatus. Antipodals, again wrong. So basically, your only answer for this particular question is what? A. Yes. Again, am I explaining? I'm so sorry. Yes. Question number 8. In Viola, both autogamy and geitonogamy is prevented. Viola. Commelina oxalis, Viola. Cow. Remember Chasmogamous and Clistogamous flowers? Here it is said that in Viola, both autogamy and geitonogamy is prevented. What do you think? So, just give me a second. Okay, so we are right over here. Yeah. So Viola, my dear children, here in this particular case, listen to me very carefully. So there can be open flowers which are something like this, right? And you have autogamy. Autogamy means self-pollination. So that is definitely going to happen. What about geitonogamy? Geitonogamy means, suppose this is a flower, there is another flower on the same plant. So from here to here, if things are going from the same plant, 2 different flowers are undergoing pollination, this is known as geitonogamy. And here it is written in Viola, both autogamy and geitonogamy is prevented. It's wrong, right? It's wrong because it can happen. Viola has closed flowers and also open flowers. Open flowers can do all this thing. Even in closed flowers, there's only one way for fertilization or pollination, pollination, and that is autogamy. So autogamy cannot be anyway prevented. So this particular statement is wrong. Now statement number 2, geitonogamy is similar to autogamy since pollen grains come from the same plant. Is it like that? So in autogamy, again, in the same plant, okay, in the same flower it is happening. In geitonogamy, same plant, different flowers. But anyway, same plant means same type of genetics are going to happen. Gametes are going to be male and female gametes. Both of them have the same genetics, right? So basically, can I say genetically it is similar? Yes. Geitonogamy is similar to autogamy since pollen grains comes from the same plant? Yes. Genetically, actually, it is similar. If at all they had mentioned that also, it would have been nice. But please read the options really, really carefully. Sometimes they could twist and turn and they could give you something else which is actually wrong. Okay, so here it is correct. it is similar to autogamy? Because they are mentioning what is the reason for that. Pull and grains are coming from the same reason. They are not saying geitonogamy and autogamy are same. They are saying it's similar, not same. You get it? So there is some differences, but there is some similarity as well. That is what you mean by that. So this particular statement is correct and the other one is wrong. So I'll say statement 1 is incorrect and statement 2 is correct. What do you mean by xenogamy? Basically, geitonogamy means 2 flowers of the same plant. Xenogamy means 2 separate plants itself. So xenogamy and geitonogamy, both of them are cross pollination, but which one of them is true? It is that one that is absolutely true. Got it? Yes. Fine. Next thing, the following picture is an example of. This particular picture is an example of what? This is given in your NCERT and we should know. This is given in a question bank also. Any idea? So over here, my dear children, you need to understand one very important thing. Okay, there are so many gynoecium, as in female reproductive part. You call the gynoecium as carpel, you call this as pistil also. So I'll say there are multiple carpels over here or multiple pistils, but generally we use the term carpels. Okay, so there are multiple carpels over here, and there is one very important thing. All the carpels, okay, all the carpels which are there, they has a unified ovary. Okay, unified ovary. So their ovary is all fused together, my dear children. And you call this condition as syncarpous condition. When they are fused, you call it syncarpous. There is a condition where there are multiple carpels, but all of them have their individual ovary. That is known as apocarpous condition. But over here, it is syncarpous that is being shown. Okay, syncarpous condition. What is this particular plant? Do you know? This is a plant from where you get what do you call opioids such as heroin and morphine and things like that. The name of this particular plant is Papaver. Papaver somniferum. This is having multicarpal condition and also syncarpous gynoecium. So ma'am, is there a condition like apocarpous as you told? Is the example given in the NCERT? Yes. That example is of Michelia. Have you seen Chambagam? In Malayalam we say Chambagam. In Hindi we say Champak. Okay. So it smells so good, the white color flowers. They have apocarpous condition and that is known as Michelia. And that is exactly known as Michelia. Okay. Got it? So that is apocarpous. This is syncarpous. Do not forget it. Okay. Fine. Next one. Yeah, so I have given the picture also. That's me. Okay, so this is Papaver somniferum. This is how exactly it looks. Okay, exactly this is how it is going to be looking, my dear children. Inside that they had the fused ovary. And over here, if at all you see, in the case of Chembagam or Champak, if you open this, it looks like this. Now this is the picture that is given in NCERT. Over here you have multiple carpels and each of them have the individual ovary. They are not fused. Okay, so this is known as apocarpous condition. Yes. Okay. Oh my God, we have completed one chapter. Kudos to everyone who have come this far. And my dear children, I hope you are understanding. Hope you are enjoying as well. So it's like more of like a drama from my end because I'm alone in the studio and I'm talking to you and I'm trying my best to make you and me highly engaged. So if at all you think it is working well for you, do not forget to share it with your friends and do not forget to hit the like button as well. Yeah? Yes. So human reproduction is the next chapter and let's see the first question that we have from this chapter. The secondary oocyte formation from primary oocyte occurs in which of the follicle? This is MQP 1 question. Okay, question number 2. MQP means model question paper. Okay. So secondary oocyte formation from primary oocyte occurs in which of the follicle? So only at 2 places the division takes place, or basically I can just say 1 place. Okay, 1 place. So you know primary follicle is there. Primary follicle is there. Inside the primary follicle, what do you have? You have the primary oocyte. Secondary follicle is being formed. In the secondary oocyte also, what do you have? You have the primary oocyte only. It has not completed its division. Okay, primary oocyte which is there, it has to divide into 2 things. It is an unequal meiotic division where you get the first polar body and you get the secondary oocyte. The secondary oocyte, that is what you have to get. Okay, so this particular transition is happening, but the division did not complete. Okay, completely the meiosis was not able to get completed off. It is stuck in meiotic phase, meiosis 1. Okay, now what happens? The secondary follicle will transform into tertiary follicle, and this is the place where everything changes up. Why do you think everything changes up? Because finally the primary oocyte is able to complete its division and form what? The secondary oocyte. finally the secondary oocyte is going to be formed up over here, and that is super important. Okay, now the tertiary follicle which is there, it is going to transform itself into the mature form of follicle which is known as Graafian follicle. And the Graafian follicle, what do you think? This will undergo division. So now the secondary oocyte is yet to divide and form the second polar body, and also one thing that is known as ovum. But this division also does not take place, so it is still stuck in secondary oocyte phase. When do you think this division is going to take place? When ovum is going to be formed? Just before fertilization. Okay, just before fertilization the ovum is going to be formed, and the sperm and the ovum is going to fuse. Okay, so always remember, tertiary follicle is the place where finally it is able to divide to secondary oocyte. So if the question is secondary oocyte formation from the primary oocyte occurs in which other follicle? It occurs in the tertiary follicle. I hope you never forget that. Yeah, in ovulation, in which form is it going out? The Graafian follicle contains the secondary oocyte. So secondary oocyte is something that is going to be released. Okay, it has not completed its division to form these two things. It happens just before fertilization. Okay. Now next thing, the cells that secrete testicular hormones or androgens are known as. The cells that secrete testicular hormones, do you have any idea? So in the testes, you have testicular lobules. In the testicular lobules, you have 2 to 3 highly coiled seminiferous tubules. If at all you zoom it in, it looks like this. So this is seminiferous tubule, this is seminiferous tubule 1, this is seminiferous tubule 2. In between this, do you see a space? Do you see the space? This space, my dear children, is known as interstitial space. And over here you have interstitial cells, or you also call them as Leydig cells. Leydig cells. What do they release? They release the androgens, the male sex hormones. Why? Because under the influence of these androgens, finally sperms are going to be produced over here in the seminiferous tubule. So in the interstitial space, you have the Leydig cells that are going to help you with all of this thing. Okay, so please remember that. Here you have, in the seminiferous tubule, you have spermatogonia. Apart from that is the sperm mother cell. Apart from that, you have the Sertoli cells, the sperm, nurse cells out there, which is there for nourishment of the spermatogonia and the sperms. So please remember, both SS cells are inside, and in the Leydig, in the interstitial space, you have just the Leydig cell. Works? Works, man. Okay. Now, yes, next question. Which of the following statement is true? Okay. LH surge induces menstruation. What do you think? LH surge means increase of LH is going to induce menstruation. No. LH surge is responsible for one very important thing. Here, always imagine the follicles are maturing and you have one Graafian follicle which is out there. This Graafian follicle has what? It has the secondary oocyte out there. Under the influence of high amount of LH, luteinizing hormone, the follicle is going to rupture and this particular guy is going to come out. Secondary oocyte is going to come out, and that process is known as ovulation. Here it is written, menstruation is induced by LH surge. Wrong statement. Now, regression of corpus luteum increases progesterone. Regression means what? Basically, the corpus luteum is going to decline. The amount of it is going to regress. It is going to be less. Do you think that is going to increase progesterone? First of all, let's talk logically. Okay, so once LH surge has taken place, this was the Graafian follicle first. It was the Graafian follicle, agreed? This Graafian, whatever is left behind, is now known as corpus luteum. Is now known as corpus luteum. So the egg is released, the corpus luteum is being made also. Not the egg, actually secondary oocyte is being released. Now what are you waiting for? You are waiting for the sperm to come. If at all the sperm and the egg comes in contact, what is going to happen? Basically, if suppose fertilization happens, you will form the zygote, then the embryo. The embryo needs nourishment. How do you provide the embryo nourishment? By the help of endometrium. Right, so you have to make sure the endometrium is super ready. Okay, ovulation has taken place. Now fertilization can happen at any point of time, but you also have to make sure endometrium is super ready. Who makes sure that? The corpus luteum, that is whatever is left behind the Graafian follicle, that forms the corpus luteum, and the corpus luteum is going to release two very important things. One is progesterone and the other one is estrogen. But this is the progesterone that majorly helps in what? Helps in, in making the endometrium really thicken. Okay, and the endometrium starts to secrete some very important things, such that if embryo comes in contact, basically it provides all the nourishment that is needed, right? This is what usually happens. But you need to understand one thing, that corpus luteum is also being maintained because there is LH being maintained. There is LH surge that happened, later LH, it comes to this particular phase where it is there, and is there for the nourishment of corpus luteum. Not nourishment, basically for making sure that the corpus luteum stays like that. The corpus luteum in turn produces what? Progesterone. But there is a catch. Progesterone, when it is produced, it does not like LH to be there, which is why it tells the papa of LH, okay, papa of LH to stop the secretion. Papa of LH is what? GnRH, gonadotropin releasing hormone from hypothalamus. So it says that stop the secretion of LH. Now if LH level goes down, that means corpus luteum is also going to go down. If corpus luteum goes down, progesterone will also be less. So regression of corpus luteum increases progesterone? No. If corpus luteum is going less, that means progesterone is also going to go less. So this is wrong. Okay. Diploid egg is formed after second meiotic division. Listen, over here, there is no diploid thing that is going to be formed. Egg cannot be diploid. First of all, egg and the sperm has to be haploid in nature. These haploid thing comes together to form a diploid zygote. So this particular statement is anyway going to be wrong. Now, embryo differentiate from the inner cell mass of the blastocyst. All of it, this question, if at all you see, you need an understanding of multiple things. Menstrual cycle, you need an understanding of how the oogenesis takes place, and you need an understanding of how basically gestation takes place, or I can say implantation takes place. So there are three different concepts being asked over here. That means we have to be really strong with our concepts. So embryo differentiates from the inner cell mass of the blastocyst. Always remember, when I say the zygote transforms into an embryo and the embryo comes and attaches to the endometrium, I mean that there is a particular stage that comes and attaches, and this stage is known as blastocyst stage. In the blastocyst stage, there are two types of layers. There is the outer layer of cells, which you call as trophoblast, and there is inner cell mass out there, or the inner mass of cells, which you call as inner cell mass. Now once it comes and attaches to the endometrium, you know endometrium cannot always provide the nourishment. There should be something out there that will be there for nourishing for a longer time, and that is a placenta. So your next goal is to make the placenta. Who helps in the formation of placenta? The trophoblast, my dear children. It helps in the formation of placenta along with the mother's maternal tissue out there. So from embryo side, it is trophoblast that is going to help. From the mother's side, it is going to be some maternal tissues that are going to help. Now what is left behind? The inner cell mass is left behind. This actually forms the embryo. This one actually forms the embryo. Rest is there for placenta formation as well. So embryo differentiate from the inner cell mass of the blastocyst. The answer is yes. This is actually correct. Sometimes you say that inner cell mass forms the embryo proper. So do not be worried. It means that it is actually the one that forms the embryo later on. Got it? Yes. So only the option is correct. Rest all of it, my dear children, are wrong. Next question. Scrotum helps in maintaining the temperature of testis, which is lower than the normal body temperature. How much degree lower than the normal body temperature? 2 to 2.5 degrees Celsius. So how many options do you have which are correct? This and this. So can I exclude this? Yes. Just cut off the options which you think that is not correct. Okay. Now what do you have? Each of the testes has about B compartments. How many compartments are there in the testis? What do you mean by compartments? You mean team lead. I always remember like that. So how many team leads are there? Testicular lobules, there are 250 of them. Testicular lobules or team leads, there are 250 of them. So here again the options are same, so we cannot choose. Now it measures C centimeter in length. What is the length of the testis? This is what you mean by length. This is the width. Width is 2 to 3 centimeter. Length is 4 to 5 centimeters. So through 3, 4, 5. 2, 3, 4, 5. Always remember that. So over here 4 to 5 centimeter is actually length. So your answer for this particular question becomes what? B. It becomes B. Okay, do not be confused with the width and the length. Length is 4 to 5 centimeter. Cool? Yes. Fine. Next question. in spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is affected? In spermatogenesis, when sperm is being formed, if FSH hormone is not secreted from the anterior pituitary, okay, which stage is affected? What do you think so? Which stage is going to be affected? Let's see. Listen, as a basic thing, okay, there is hypothalamus. Hypothalamus produces gonadotropin releasing hormones. Okay, now these gonadotropin releasing hormones are going to act on anterior pituitary. Now the anterior pituitary, on the other hand, is going to release two things. One is LH and the other one is FSH. Cool. LH, which is there, it is going to act on the Leydig cells, and FSH, which is there, it is going to act on the Sertoli cells. Remember, Leydig cells in between the seminiferous tubules, Sertoli cells inside the seminiferous tubules. So here L, here L, here S, here S. You can remember like that. Okay, the Leydig cells, on the other hand, is going to release what? The male sex hormones, which you call as androgens. Done? Done. Okay, what about the Sertoli cells? What are they going to do over here? When they are acted by FSH, they are going to release some factors. Some factors. Okay, so two of the things are being produced. Here androgens, that is the male sex hormones, and they are the factors. You need to understand that basically this thing is happening within the seminiferous tubule. Factors are being produced within the seminiferous tubule. What is happening in the seminiferous tubule? The spermatogenesis is happening over here. Spermatogenesis is happening over here. And for that, you need high level of androgens. So the androgens gets inside. Okay, why do you think the factors are being produced over here? Factors are here for making sure that the level of androgens are being maintained. Why? Because if at all androgens are not there, this process is not going to happen. Especially this particular process. What is that process, ma'am? So in spermatogenesis, if at all you know, the spermatogonia differentiates. You have one cell that is known as the primary spermatocyte, which is in 2N nature. This primary spermatocyte undergoes meiosis. You get two cells, N and N. Okay, this is going to be what? Your secondary spermatocyte. Again, here half of the number of chromosomes have gone. Okay, only half is left. Now again meiosis happens. You get four cells. Are they the sperms? No, they are known as spermatids. Spermatids, they are like kids. They are immature kids, chotu kids. They are immature. They are globular in shape, but the actual sperm is elongated in shape. Now you need to transform this into what? This way, which means that you have to make them transform into spermatozoa. You need to make them transform into spermatozoa. So spermatids to spermatozoa transformation has a specific name. It is known as spermiogenesis. Why are you teaching this, ma'am? Why are you teaching this, ma'am? Basically, in spermiogenesis, you need to make sure the level of androgens is maintained really high. So throughout it is important, but majorly during this particular process. So in spermatogenesis, while the whole formation of spermatogenesis is happening, if FSH hormone is not being secreted from the anterior pituitary, what stage is going to be affected? Formation of primary spermatocyte? No, that is going to happen. Formation of spermatozoa? Yes, that is not going to happen, because as I said, here in the process of spermiogenesis, this whole is spermatogenesis, but this particular portion is spermiogenesis. At this particular place, you definitely need a lot of FSH, because FSH acts on the Sertoli cells to produce factors, and these factors make sure testosterone level is being maintained. So this is the thing that is being affected. Now formation of spermatids? No. Formation of secondary spermatocyte? No. Your only answer for this question is going to be B. So for this, again, you need a an in-depth understanding of spermatogenesis. Later when the sperms are being formed, the sperms will be moving out of the seminiferous tubules, and that is what you call as spermiation. I remember it like aviation. Similarly, you can also remember, but yes, spermiation means basically the sperms are being moving out. Okay? Yes, ma'am. Okay. Now, to produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? To produce 2000 sperm, remember, from one spermatogonia, as I said, it transforms into primary oocyte and things like that. At the end, you get 4 sperms. At the end, you get 4 of the sperms. So here the question is 2000 sperms are there. So 2000 divided by 4, if at all I do, I get an understanding of that there are 500 spermatogonia, which divides, each of them divides into 4 sperms. That is how I get 2000 sperms. Okay. What about oogonia, my dear children? You have been asked about oogonia. Remember, one oogonia transforms into one primary oocyte. This primary oocyte undergoes unequal division. Remember, first polar body and the secondary oocyte. This first polar body does not undergo any division. This does not undergo any division. Now only this is left. This also undergo unequal division, which is why you get 1 ovum at the end. You do not get 4 ovum and all, because here also did not undergo division. Here also did not undergo division later on. So from 1 oogonia, 1 ovum. So if at all you are saying that 400 ova is being produced, 400 divided by 1, it will be 400 oogonia only. Got it? So 500, where is it? 500 spermatogonia and 400 oogonia is going to be your answer for this question. Your answer is B. Cool? Yes. Next question. Which among the following has 23 chromosomes? That means half the number of chromosomes. How do I know that? Because I know 46 number of chromosome is what? Diploid. So half of it is going to be what? Half of it is going to be 23. And where do you see this? You see this either in the case of ovum or the sperm. Apart from that, as I said, meiosis happens in 2 stages. In the first stage itself, my dear children, what happens? Diploid turns into haploid. So basically, secondary oocyte or secondary spermatocyte, all of them are going to be haploid. Half the number of chromosomes are going to be there. So spermatogonia, definitely not. It is a diploid cell. Zygote, definitely not. It is made of fusion of haploid, diploid. Secondary oocyte, yes. Secondary oocyte, why? Because I told you, primary oocyte undergoes first meiosis. End is what? Reductional division. Okay, that means it will have half the number of chromosomes. Later, it will be carrying the same number of chromosomes in the ova. Or in the case of males, it will carry the same number of chromosomes in the case of the spermatids which are going to be formed. So it is secondary oocyte or secondary spermatocyte, which is the answer. And you will not be getting all the options together, that is secondary oocyte and also ovum. That is definitely not going to happen. Okay, you will only get either secondary oocyte or the ovum. Yeah. Okay, so next question. Identify the wrong pair with respect to menstrual cycle. Formation of corpus luteum, 15th to 28th day. So if at all I say the cycle is of 28 days, when do you expect that the ovulation is going to happen? On the 14th day. That means this is a period when there was high amount of LH. Now, after this, when the LH level goes down a bit, basically the Graafian follicle has ruptured. Now you are going to form the corpus luteum. So corpus luteum is formed after the 14th day. Formation of corpus luteum 15th to 28th day. Yes, it happens during that particular point of time. This is correct. Secretion of progesterone. Obviously, the corpus luteum is the one that releases progesterone. So I'll say again it happens during the same day, 15th to 28th day. Regeneration of the endometrium 5th to 13th day. 5th to 13th. So listen, this is a cycle. From 1st to 5th day, that is there, you will have the menstrual phase. Menstrual phase basically means that your endometrium is going to fall down. Okay, it is going to break down. Now, after the 5th day, my dear children, slowly the endometrium is going to regenerate, because again we are preparing for the next cycle of ovulation to happen, right? So regeneration of endometrium from 5th to 13th day, yes, from this particular time it happens. So this is also correct. Okay, disintegration of endometrium 5th to 13th day. Okay, this was correct, this was correct. Disintegration of endometrium. Disintegration means again breakdown of endometrium from 5th to 13th day. No, regeneration happens Disintegration took place over here in the 1st to 5th day. After that, again it is being formed. Ovulation happens. It becomes even more thicker. And then when, if suppose fertilization didn't take place and things like that, again it will fall down. So here it is time for regeneration. It is time for regeneration. Got it? And here it's a time for thickening. So here it is written ulta. So this is wrong. Disintegration of endometrium from 5th to 13th day. No, it happens in the 1st to 5th day. Yes? Yes, ma'am. Okay. Next question. The secondary oocyte after ovulation is covered by non-cellular layer of what? Listen to me. It's not like after ovulation, but in the graphene follicle, I told you there is secondary oocyte which has not completed its division, right? This secondary oocyte which is there in the graphene follicle makes a layer around it. And there's a unique feature about this particular layer. It's non-cellular. That means there are no cells up over here, or I'll call this as a cellular layer. It is not made up of any cell out there. You call this as zona pellucida. You call this as zona pellucida. So C is your answer for this particular question. Okay, later around it, since it is there in the graphene follicle, no, the cells of the graphene follicle, they also contribute to one more layer formation, and that is known as corona radiata. That is cellular, and that is known as corona radiata. This is how it is going to be released from the graphene follicle with all these things. Okay, so non-cellular layer is there, that is known as zona pellucida. It is acellular also, please remember. Cool? Yes. Next, during pregnancy, the fetus develops limbs and digits after what? Limbs and digits. This is a very important question, as in they can give you any month and you will be asked this question, so you should know. So during pregnancy, the fetus develop limbs and digits. So first month, the first thing that we check, okay. First month, the first thing that we check when someone dies is what? Heartbeat. Similarly, when someone born, we check the heartbeat. So towards the end of first month, you have the heart being formed. Now the second month, my dear children, you have the limbs and also the digits being formed. Limbs and the digits are going to be formed. They are not going to be separate. They might be together, but they are going to be formed. By the third month, my dear children, you will be having all of this definitely there. Apart from that, external genital organ. That means male or female external genital organ will be there. Apart from that, the organ and the organ system, most of them are going to be formed over here. Organ and the organ system, most of them are going to be formed here itself. Okay. Now, fourth month is not given in your NCERT. Until third month, we say first 3 months is known as the first trimester. And always remember, 1 month is equals to 4 weeks. In 1 month, how many weeks do we have? 4 weeks. So 3 month means 3 into 4. 3 into 4 is equals to what? 12 weeks. So by the end of 12 weeks, what do you have? You have all this thing being formed. So they can ask you in different format. Just know that. Okay. Now the 4th month is skipped. 5th month. Baby says that, God, basically I want to move around. Until now I heard you. Okay. But now I want to move around. So you see the first movement of the fetus in that month. And also one more thing, baby says that I want some hair to do some fashion show. So over here it has the hair on its head, hair on the head, and also the first movement in the 5th month. Now in the 6th month, that means the next 3 months, in the 6th month, my dear children, baby asked the God for hair, no? So God gives lots of hair on the body also. So you will see tiny hair on the body. Apart from that, eyelashes are going to be formed. And my dear children, eyelids are going to be separated. These are the few things that you can clearly see. This is basically the second trimester. This was the first trimester. This is going to be the second trimester. You also call this as 24 weeks. Why? Because 6 into 4. 6 months. Okay, 6 months into 4, 24 weeks as well. Just know that. Here the question is, during pregnancy, the fetus develops limbs and digits. Limbs and digits, in the second month means 2 into 4, 8 weeks. So after 8 weeks. Okay? Yes. Okay. Next question. Statement number 1. The process of release of sperms from the seminiferous tubule is known as spermatogenesis. What do you think is the answer? The process of release of sperms? Arey Baba, release of sperms. Remember, aviation, spermiation. Spermatogenesis is formation of sperm, right? So this is wrong. the spermatids are transformed into spermatozoa, is it? Or is it spermatozoa to spermatids? Remember, kids to zoa. Okay, so it was immature, now it is being mature by the process of spermiogenesis. Is that correct? Yes, that process is known as spermiogenesis, and here you need a lot of factors from Sertoli cells. Remember? Yes. So statement 1 is wrong, statement 2 is correct. Fine. Next question. Choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. Okay. Zona pellucida is the thing that is being labeled over here. So you have to say about the formation and also the dissolution, as in breakdown and also making. So how it is made? It is made with the help of secondary oocyte. I told you it makes. So there are two options. I will cancel out these two options. Now, dissolution as in breakdown of zona pellucida. When does it happen? Before fertilization, after fertilization. Before fertilization, do you think zona pellucida will go away? Why do you think zona pellucida is there at the first scenario? You need to understand that. Basically, zona pellucida has some receptors onto which your sperm head is going to attach and going to release some enzymes such that it can cleave through and get entry. Once a sperm enters, what is going to happen? The zona pellucida is going to become very, very thick such that no other sperms can enter. Okay, later fertilization is going to happen. So for fertilization to happen, this guy has to be here. So it cannot be before fertilization. Now after fertilization has taken place, after zygote is being formed, slowly the zona pellucida is going to be going off. So after fertilization, it gets dissoluted or basically dissolved. Okay, yes. So your answer for this question is going to be D. Every time in an MCQ question, read the question carefully. Very, very important. Most of us mess up with the MCQ question because we might know the answer, but we are in an anxious state such that we read it very, very fast. So don't do that mistake at all. Okay, very, very important thing. Now, so I think with this we completed your human reproduction chapter, and with this I want to tell you one very important thing. That is, my dear children, we have launched a KCET crash course, which is 30-day comprehensive crash course for everyone who is preparing for KCET. Now I know not a lot of biology students are in for KCET examination, but still I am telling you, if at all you have not started up your preparation really well or strong, we are here with you. There will be complete PU1 and PU2 syllabus coverage. There will be previous year questions along with the solutions. There will be PYQs in the form of test also, that suppose you're done with the chapter, you can go give the test of that particular chapter. There will be 10 full-length mock tests, and all of it starts from 21st of March, just after your board examination finishes. Yeah, and the pricing for this is rupees 2499, but it will not be the same, okay, after 25th of February. So if at all you're planning to take it up, make sure you enroll right now. Yes, and if you want a free mock test PDF, just chumma, okay, if at all someone is interested, there, go to the link. Okay, go to the description section. I somehow not able to tell that. Okay, and then click on the link, fill in your number and download the mock test. But as of now, I don't think you will require it, but check it out. Okay, definitely check it out. Okay, now we go ahead the next chapter, that is reproductive health. And just give me a second, let me go have some water. You also, every one of you who is just watching this video and religiously following everything that I am telling you, please you also drink some water, have some snacks also, and come back. Okay? Yes. You're champions. So did you drink water? I hope you're doing great. And by the way, everyone who have reached until this particular point, let me know in the comment section that ma'am, level 1 crossed. I will be really happy to see that. Actually, I'll be really happy when you cross the last level out there, yeah. But still, you can keep on updating in the comment itself. I'll be really happy with that. I said so many times, really. Okay, so basically, I just got a headache and I went and put in so much balm around all different parts of my face, which is why I'm not even able to open my eyes now. Okay, so yes, let's go ahead with the first question that we have, and this is from your MQPs, what I believe. Yes. So oral pills as contraceptive acts as contraceptive effectively. Oral pills as contraceptive effectively functions by inhibiting what? Okay. So oral pills, when I say that they are contraceptives, how does it show their effect? How they are actually doing their effect? What do they inhibit? And maybe acid, richness, nostoc, ovulation, colostrum, diversity. So you all know it's basically going to be ovulation itself. But why do you think that oral pills are going to inhibit ovulation? Listen, oral pills has a combination of progesterone, or estrogen together. Otherwise, it can be plain progesterone and estrogen alone as well. Anyways, these hormones which we see are generally required for the process of ovulation and also for the maintenance of endometrium. Now, when you are providing it from outside, basically that is going to create what? An imbalance. And since there is an imbalance that is going to happen, it will do the opposite of what it was doing. So since usually it helped in ovulation, now it won't. Usually it helps in formation of thick endometrium, now it is not going to be forming thick endometrium. Instead, you will be having thin endometrium being formed, and this thin endometrium cannot support, cannot support any fetus out there, which is why obviously pregnancy will not be happening. So that is how usually it works. So oral pills act here by inhibiting what? Ovulation. Do you know, your ma'am is this brilliant that she almost put the balm inside, the roll-on balm inside the eyes. Oh my god. Yes. Now, which of the following is not a copper releasing IUD? You have to spot that. Which is not? Children sometimes what they do is that they try to find that which one is copper releasing. So please know it is asking about which one is not. Okay. LNG20, QT is copper T basically, and copper T is copper releasing only. Cu7 is again copper releasing. So here Cu2T looks like this, onto which you have the copper round. Cu7 looks like this, onto which you have the copper round. That is the only difference. Now multi-load 375, what about that? This is also copper releasing IUT, and over here you have 375 mm or something like that of copper being releasing, which is why not mm, some unit was there, which is why you call this as multi-load 375. So this is also copper releasing. So what is left behind? LNG20. LNG20 is levonorgestrel. Basically it's a synthetic form of progesterone, my dear children. It is coming under hormone releasing IUD. Okay, and there is hormone releasing IUD, there is copper releasing IUD, there is non-medicated IUDs, right? So this comes under hormone releasing IUD. It functions similarly just like how oral pills were working, because over here also whenever you're giving synthetic hormones, for example progesterone, it creates an imbalance leading to ovulation not happening, or thin endometrium being formed, or the cervix being cervix having one mucus around it, mucus cap around it, such that the sperms cannot move inside. So that is a general thing that is there. So this is hormone releasing, and 3 of them are copper releasing. Copper, what does it do? It messes up with the motility of the sperm and also the fertilizing capacity of the sperm. So copper, they go and stick like to the sperm's end and say that you cannot move. That's how it works. Okay, so you can remember like that if at all you want. Yes. Now, statement number 3, question number 3, statement number 1, GIFT is an in vitro fertilization technique. Is it? GIFT means gamete intrafallopian tube transfer. Do you think it's an in vitro fertilization technique? In vitro means what? In vitro means inside the laboratory. GIFT is where you take the gametes and you transfer the gametes into the fallopian tube. Why? Suppose there is a female X. Okay. She is having a proper uterus, everything, but she is not producing viable types of ovum, or a ovum which is there is not of good quality, which is why fertilization is not happening. Rest everything is really cool up there. So basically in GIFT, what do? Some other lady who has a normal ovum will be gifting her ovum. That is what gamete. Okay, gamete is being gifted into what? Inside the fallopian tube, it is being transferred as well. This particular gamete that is being gifted is being transferred into the fallopian tube. Why? Because you are relying on your own body for fertilization to happen. The sperm will be coming by natural sexual intercourse, and the sperm will be basically fertilizing the ovum out there. So here it is not in vitro, rather it is in vivo. Opposite of it. It's inside the body, naturally occurring. So this is a wrong statement. Statement number 2. In GIFT, gametes are fertilized outside the female body. Are the same thing is being said? Okay, do you think gametes are fertilized outside the female body? Is it? Gametes are fertilized inside the female body. That is why I told you it's in vivo, not in vitro. So both the statements are wrong, but I do not know what I have marked. Yes. So statement 1 and statement 2, both of them are wrong. Incorrect. Okay? Yes. Next question. Out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. Out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. When do you transfer the embryo? So, in vitro fertilization, that means fertilization in the laboratory happens. After that, you get the zygote. This zygote can be single cell, right? Or later it can divide to form the embryo. Now embryo up to 8 cell stage, okay, can be transferred into the fallopian tube. Here you have been asked about that only. Up to how many stage you can transfer into the fallopian tube? It's only up to 8 blastomeres or 8 cell stage. The cells are known as blastomeres. Now if it is beyond 8, you cannot put it in the fallopian tube. Why? Because ectopic pregnancy can happen. That means in the fallopian tube the baby can be formed, and that can be dangerous for the mother as well as child. Not can, it will be dangerous for the mother and the child. So embryo which is up to 8 blastomeres can be put in the fallopian tube. If at all it is beyond that, you have to put it in the uterus. It is known as intrauterine transfer. In that case, you call this as intrauterine, inside the uterus transfer, intrauterine transfer. In this particular case, you call this as ZIFT, zygote intrafallopian transfer. So it is not zygote, it can be an embryo up to 8 blastomeres also. So either zygote or an embryo up to 8 blastomeres is transferred into the fallopian tube. Works? Works, ma'am. Okay. So your answer for this question is A. Now, reasons for human population explosions are given below. Reason why human population is increasing is given below, and you have to select the correct answer statements from the options. Okay. Rapid decline in maternal mortality rate. Mothers are dying less, which is why population is increasing. Yes, mothers are dying. There's a proper care being given. It's a good thing only. Okay. So this is definitely helping. Now rapid decline in infant mortality rate. Again, the babies are also dying less, which is why the population is also stable or increasing. Now, rapid increase in death rate. If people are dying more, do you think it will lead to population increase? No, Baba, it will lead to population decrease, right? So this is wrong, because here it is written rapid increase in death rate is leading to population explosion. No, that cannot happen anyways, right? Now, increase in the number of people in the reproducible age. Again, if people are there who are going to reproduce in the future and their number is more, obviously the population explosion is going to happen. So I'll say 1, 2 and 4 statements are correct. 1, 2 and 4 statement, that means your answer is going to be C, Baba, right? Your answer is going to be C. Yeah? Yes. Now the next question that we have is question number 6. Which of the following sexually transmitted diseases is not completely curable? So there are some STDs or venereal disease or genetic tract infections which can be treated, which can be cured also, but 3 of them are there which cannot be cured, and 3 of them has H in it. One is HIV AIDS, other one is genital herpes, and the third one is hepatitis B. Hepatitis B. Out of this, if at all you see any of these options, your answer is that. Man, there is no HIV, there is no herpes, there is no hepatitis B. You see genital herpes, that is itself the herpes. So your answer for this question is going to be C, Baba, which is not completely curative. Yeah? Next, an example for non-medicated IUD is non-medicated IUD. That means this is an intrauterine device. You put it inside the uterus only, but it is not going to release anything in your body. It's not going to release any hormones or medicines in your body. What is that? So CUT, copper T is releasing copper. LNG is reducing levonorgestrel, the synthetic form of progesterone. Multilode 375 is also releasing Cu, that is copper ions. Lipase loop is there, which is going to be the answer. It is the example for non-medicated IUD. So basically, suppose this is the uterus. In the uterus, the shape of the uterus is like this. Similarly, you put something like this over here, which looks like a loop. This is known as lipase loop. Okay, and it is not there to release anything. Basically, when something foreign is being put in our body, what happens? Our body reacts to it by producing immune cells, okay, that fight against the pathogens. Now suppose the sperm enters the cell, enters our body. What is going to happen? Our immune cells feel that this guy is also a problematic guy, so we will kill them. Phagocytosis is going to happen. The cells are going to be killed. They're going to be taken in and be killed by the immune cells out there. Yeah, so lipase loop is there for generally activating our immune system is what I'll say. Yeah. Now the assisted reproductive technologies in which a sperm is directly injected into the ovum. Sperm is injected into the ovum. Why do you do that? In the laboratory condition for fertilization to happen, right? What do you call this process as? You have a specific name. Remember, do not mug it up. This is the egg. This is the cytoplasm of the egg. And suppose you are putting in the sperm with the help of a pipette. You call this as intra, that means inside. Inside what? Intracytoplasmic. Inside the cytoplasm, what you are doing? You are injecting the sperm. So sperm injection. Intracytoplasmic sperm injection. So your answer for this question is ICSI. Intracytoplasmic sperm injection. In this particular part of the chapter, if at all you go around mugging things, you will be forgetting it. So please think about it and you will get your answers. Yeah? Yes. Next, coitus must be avoided from day 10 to day 17 of the menstrual cycle. The reason is, okay, this is a natural method of contraception, and it is said that it does not have any side effects, but it has high failure rates. Yes. So why do you think coitus or sexual intercourse is being avoided from day 10 to day 17? Because, my dear children, I told you, if your cycle is of 1 to 28 days, the mid cycle is 14th day where ovulation can happen. But we are not sure that ovulation will happen on 14th day. It can happen over here. It can happen until here also. So 10 to 17 days, I'll say it's the fertile window where the egg can release any single time. And always remember, suppose the egg is not released, but the sperm is being put inside your body. That means sexual intercourse have taken place. Sperm after ejaculation can stay in your body for 5 days. That means 72 hours. Now, later on, if the egg is being released, the sperm is still there, they can fuse, you get it, which is why we say that it should be avoided. So sperm remain viable for 7 days. Ovum may be available for fertilization. Yes, that is the reason. Ovum may be available for fertilization because that is the ovulation period, that is a fertile period that is out there. Okay? Yes. So if at all, yeah. So ovum loses viability on these days, none of these, no. So I hope you you got to understand the answer. Why 10 to 17 days? Because here you're specifically talking about ovum. Ovum could be available. Sperm though, by any time it can be available, but ovum is available only during these days. Okay? Yes. Now, yeah. Condoms have additional benefit as compared to other contraceptive because, what do you think? Additional benefit as compared to other contraceptive because what? What is the reason for that? Or what does it do? It is made up of rubber and latex. Other things are also made up of rubber and latex. You have so many things made up of that. For example, diaphragm and things like that. Okay. Provides privacy to a user. Almost all the things provides privacy to the user. Protects against STIs, sexually transmitted infection. Do you think that is the additional benefit of having a condom? Yes. Because when you use a condom, suppose it is a penis, it is being covered by a condom. Any genital fluid is trapped over here in the condom itself. Genital fluids are not being exchanged, and if genital fluids or blood, anything is not being exchanged, you say that there are lower chances of getting an STI. You get it? So it protects against STI, that is the best part. It prevents implantation, no baba. That is a whole different thing. So C is your answer. It prevents against STI. That is an additional benefit that you get from condoms. And that is the only one that can provide that benefit. Now, there's a statutory ban on amniocentesis as. Why there's a ban on amniocentesis? It reveals the chromosomal anomalies. Chromosomal anomalies means problems in the chromosome. If it reveals, it's actually good. No, we get to understand that, right? And we can treat it. So that's not a reason. It can be used for determining the sex of the unborn. It can be used to reverse the sex of the fetus. No, obviously not. It can be used for correcting the genetic disorders. Yes, it can be. But do you think it has been put a ban on it because of the sole reason? No. The reason is that basically it can be used for determining the sex of the child as well. And when they get to understand that it's a female child, they go ahead with female feticide. That is why there was a ban being put on amniocentesis. So always remember, amniocentesis were actually found out for finding the genetic disorders out there. And by genetic disorders, I mean hemophilia, sickle cell anemia, Down syndrome and things like that. Not jaundice. Jaundice is not sickle, but you call, it's not a genetic disorder. Okay. And apart from that, I can also find the sex of the child, but that was not the primary reason for inventing amniocentesis, right? The primary reason was for finding genetic disorders, but people went ahead, found the sex of the child and killed when it was a baby girl. So that was a very, very big problem. So that's why a ban was being put. Okay, yes. Now, select the options with correct identification of the given contraceptive measure and the hormone essentially present in it. So this is the contraceptive thing that is being there. And what do you think is the, yeah. So is it an IUD, intrauterine device? Do you put it inside the uterus? No. Everything that you put inside the uterus looks like the uterus cavity is. Some shape would be there like that. What about CUT? Copper T is definitely not the answer. That is IUD, right? And subcutaneous implant, progesterone and progesterone estrogen combination. Intramuscular implant. What do you think this is? This is a subcutaneous implant. It's an implant which you put it below under the skin. What does it release? Over here it is said you have to find the name of the given contraceptive measure and also the hormones essentially present in it. So the hormone is what? Progesterone or progesterone estrogen combinations. So subcutaneous implant And apart from that, it is what? It's a combination of progesterone or progesterone estrogen combinations out there. Okay, and again it does the same thing just like oral pills, prevents ovulation, and it will be not allowing the endometrium to be thickening up. There will be cervical mucus thing that is going to be formed. It does not allow the sperm to move around. So always remember that. Yeah. Next thing, a lady is 8 weeks pregnant. 8 weeks pregnant. 8 weeks means how many months? 8 weeks means how many months? 2 months, right? Okay. Various tests done confirm that if a child is born, it would suffer from a serious illness. What is the best suggested option in this particular case? It's been 2 months only, and the baby might suffer from a serious illness. What could be done? Amniocentesis is to be performed, emergency contraceptive pills is to be taken, MTP is to be performed, tubectomy is to be performed. Listen, probably amniocentesis might already been performed. That is why you say that the child is suffering from the serious illness. The next thing that you should be doing is MTP, medical termination of pregnancy. This has to be done. Medical termination of pregnancy has to be done because you know that the child, if at all it is born, would have a serious illness. So that is a thing, and it's just 2 months. So it is definitely safer for the person also. Just a second, okay? Over here I have written amniocentesis is to be performed. Just give me a second. I'll be back. Okay, so I just changed the color of the answer so that when you get the PDF, it's not wrong. Okay, so it is MTP is to be performed, not amniocentesis, because with the help of amniocentesis, obviously you have found that serious illness is there, or basically with some other method also you could have found that serious illness is there. Now what is the best suggested option in this case? The next thing that you should be doing is what? MTP has to be performed. That is the next thing. Okay, medical termination of pregnancy. Cool. Now, which of the following contraceptive devices is having a common mechanism of action? Has the same mechanism of action? Valves and condoms, both of them, they are physical barriers. CUT and multilode, both of them are copper releasing IUDs. Progestasert as well as cervical cap. Progestasert as well as cervical caps, definitely not, because cervical cap is a physical barrier, and progestasert is, my dear children, something that is related to hormone releasing thing, right? So not. Mala-D and lipase loop. What do you think is the answer for this particular question? Please think about it and then only tell the answer. Think very nicely. It is this. Why? Because valves and condoms, they are physical barriers, but valves access a specific location. Condom, my dear children, again over here you have a whole coverage. In the case of valves, it is kind of different. Okay, there might be vaginal valves and things like that. This used to cover the vagina. Now condom is not like that. It can be used in the case of males as well as females, and it has a different way. It also protects against STIs as well. So there is a difference. When you talk about CUT and multilode 375, both of them has a common function, and that is to produce what? Copper ions. Copper ions are going to mess around these sperm motility as well as the fertilizing capacity, which is why this would be more correct. Mala-D is not there in your NCERT, which is why you should not even consider that. So yes, that is the only answer that you need to have. Cool? Yes. Next question. If a female has rudimentary ovary but can provide a suitable environment for the growth of the fetus, the ART technique or the ART, assisted reproductive technique, that can be used is, so here she has a rudimentary ovary. That means her ovary cannot produce enough amount of good ovum. Okay, her rudimentary ovary means underdeveloped ovary, might not be producing ovum itself. That can also be a case. Now in this particular condition, you need to notice one more very important thing, that there is she can provide a suitable environment for the growth of the fetus. That means uterus and everything is fine. What is the best assisted reproductive technology that can be suggested over here? It can be IVF-ET. IVF-ET means what? In vitro fertilization embryo transfer. Embryo, why do you need to transfer the embryo directly? You can just give the egg because she cannot produce the egg, right? So it is going to be gamete intrafallopian transfer, GIFT. Just provide the gamete, rest everything is fine. Not artificial insemination, because in artificial insemination, what do? You put the semen inside the vaginal area or inside the reproductive tract. If at all inside the reproductive tract, you will call this as intrauterine insemination. If in the vaginal area, generally you say artificial insemination. But that is the case when basically the sperms are naturally not going ahead and doing the fertilization process, or when the motility is less, you take it from the donor and you provide it. So it happens in that particular condition. Okay. Now CUT and multilode 375. What about this? No. There's only one answer, that is the straight question. Cool? Yes ma'am. Cool. Now we are going ahead with the next chapter, but before I go ahead with that, I just want to tell you one more very important thing. Until when is it safe to do MTP? It is safe to do MTP until 3 months, and by 3 months I mean 12 weeks. But until what time can it be done? That is also a question. So until what time it can be done? It can be done until 24 weeks. That means 6 months. But until what it is safe? Okay, very, very important. Do not mess around over here. Got it? Yes. And yeah. In which year MTP was legalized? And in which year MTP Amendment Act was being put? So please know about that. MTP was legalized in 1971 is what I remember. Okay, and basically if at all you add +1 over here, it becomes 20, and here if at all you do ulta, it becomes 17. So in 2017 the MTP Amendment Act was being put down, according to which if at all a person is performing MTP at this particular time, you need the permission from at least one medical practitioner, and if at all 6 months, then you need the permission from 2 medical practitioners. That is how it goes. Okay, so please know about that also. Now we go ahead to the next chapter, that is molecular basis of inheritance. So my dear children, before I go ahead with that, I always want to tell you one very important thing. It's not like any teacher who is in YouTube, who is in school, anyone who says that this particular questions are something that is going to come, you need to understand that they mean that the concepts could be repeated, the questions will not. So your concepts have to be strong. Don't think that just by relying on questions your whole thing is done. No, you have to make sure your concepts are strong. We have recorded masterstroke videos. Watch it if at all you need. Make your concept super clear, then go ahead with question practice. This is important. If at all you're doing question practice, do that, but along with that your concept clarity is also important. Okay? Yes. Now you have molecular basis of inheritance, and you only get one question from this chapter, one MCQ, which is why I'm not taking much, but yes. Okay, let's see. So another chemical name for the nitrogenous base thymine is already also known as what? So listen to me. In the case of DNA, you have thymine as a pyrimidine base. Okay, so in DNA what do you have? You have A, G and TC. Take care, AG, take care. Okay, so you can remember like that. But in the case of RNA, you do not have thymine, you had uracil. RNA came first or DNA came first? It was definitely RNA that came first. Later on some modification happened, that's how you got the DNA. So uracil was there firstly, then uracil got modified into something that is known as thymine now. How modification took place? At the 5th position, a methyl group was added, and that is why it is known as 5-methyl uracil. Thymine is also known as 5-methyl uracil for the same reason. On the 5th position, a methyl group is being added. Got it? Yes. Next thing, if an mRNA 3-AUG undergoes translation, how many types of amino acids are going to be formed? If an mRNA, which is like this, undergoes translation, how many types of amino acids are going to be formed, are going to be present? That is a question. Now usually the mistake that we all do is AUG, okay, methionine, then UU phenylalanine, UU phenylalanine, UU phenylalanine. So 1, then 2, this all thing is same only, so I will not count, and UAA is a stop codon, so basically that is not going to code for anything. So two types, okay, 2 types of amino acids are going to be formed. Is it 2 types of amino acid? That is there in the option, but that is not the answer. How many types of amino acids are going to be formed over here? Please understand, see the polarity, it is 3' and 5'. Translation happens from 5' to 3'. So you have to read in this particular frame. So it is AAU, one amino acid, UUU, another type of amino acid. You just give me a second. Another type of amino acid. This is UU, same type of amino acid. Similarly, UUU, same type of amino acid. So I'll say phenylalanine is the next type, and then GUA is the 3rd type. So you get 3 types of amino acids being formed over here. You get it? So B is your answer for this question. Okay, check out properly, then only go ahead. Otherwise, you will lose your marks, baba. Okay. Next, match the following: Alec Jeffrey, F. Sanger, Jacob and Monod, and Griffith. Always in match the following question, read all the options first, then only go ahead with matching of the options. Okay, and always try to match A and D first. It will be easy for you then. Alec Jeffrey was a person who went ahead and gave DNA fingerprinting method. Jeffrey has double F over here, that's how I remember that. You can remember it other ways also. Griffith, my dear children, Griffith was a famous guy for doing the transforming principle experiment. Not transferring, it is transforming principle experiment. So A goes with 4. A goes with 4. I have two options. Let me cut out these two options. And D goes with 2. A goes with 4 and D goes with 2. Here I've got my answer. But you have to be very sure your A and D options are correct. Now F. Sanger is responsible for annotated DNA sequencing. You remember whole DNA was being sequenced with the help of this technique. So that is F. Sanger who gave up this particular technique. Jacob and Monod, both of them has O. O stands for lac operon. Jacob and Monod basically were the people behind lac operon. Okay. So what is your answer? Your answer is going to be B. Next question. GGU, GGC, GGA, GGG, all code for glycine. All the 4 codons are coding for glycine. Which salient feature of this genetic code does it prove? Which feature does it prove, my dear children, of the genetic code? You have so many features of genetic code, right? There's a feature that is known as degenerate feature, where multiple codons can code for the same amino acid. So here, these are the multiple codons and they are coding for the same amino acid that is known as glycine. So this feature is known as degenerate feature, or you call this as degenerate codons. But always remember, one codon codes for only one amino acid. GGU, whatever you do will be coding for only glycine. That is known as unambiguous nature. This, when you talk about multiple codons coding for the same amino acid, you say it is degenerate nature. Done. Okay, cool. Next go. In a single-stranded DNA, if the ratio of A and G is 60 and 40, then the ratio of T and C. Whenever you have this type of question, you go like this: A and G and T and C. If A and G is 60-40, then what is going to be T and C? T and C is also going to be 60-40, because how many number of A is there, that many number of T will be there. How many of G is there, that many number of C is going to be there. That is how it goes. Is it? Remember, read the question carefully. In a single-stranded DNA, A can be multiple times, and so does T. T can be also multiple times. Here, A and T ratio is not relatable. You get it? How many numbers of A are there? But that we cannot say how many number of T is there. You get it? Because here you're talking about single-stranded DNA. So none of the above is going to be the answer for this particular question. Got it? Very, very, very important. Fine? Yes. Now, the next one. The components of deoxyguanosine is there. So, if at all you have studied my master stroke series, you will get to understand the answer for this question very, very easily. I've told. Deoxyguanosine. Deoxy means here which sugar is involved over here? Deoxyribose sugar is involved. Okay? Deoxyribose sugar, that means it's a DNA. Cool. Now, over here when I say guanosine, deoxyguanosine, I do not say deoxyguanosine triphosphate. If I would have told that, then phosphate group would have been included. But here, there is no term phosphate included. That means it is just the ribose plus the nitrogenous base, which is what? Guanine. Which is guanine. When both of them come together, the deoxyribose as well as guanine comes together, I will call this as deoxyguanosine. Okay, deoxyguanosine. Fine, so your answer for this question is going to be this. Why not this? Because here it is written phosphate, here also it is phosphate, and here it is ribose sugar, here it is deoxy mentioned, right? So it cannot be ribose sugar for sure. Got it? Okay. If at all it was plain guanosine, then what would have been your answer? If at all it was plain guanosine, what would have been your answer? Then the component will be guanine and ribose. Because for ribose, you do not call it riboguanosine. You do not call it something like that. You say guanosine only. Guanosine is basically nucleoside. That means sugar plus guanine. That is a nucleoside. And since in the case of RNA, you do not have any deoxy term, you just call it guanosine, adenosine, and cytosine and things like that. When you add deoxy, that means specifically talking about DNA. That's all. Okay? Fine. Please know that. Next, a mature processed mRNA transcribed from a gene is 636 nucleotides long. How many nucleotides are there? 636. Okay. Assuming it codes continuously without UTR and stop codons, that means there is no untranslated regions, there is no stop codons, and it is continuously being translated. How many amino acid does it encode? So 636 nucleotides are there, no stop codons, nothing. So I'll say 636 divided by 3 because 3 codons come together to form one amino acid, right? So basically your answer will be 212. 212 codons are there, 212 amino acids will be there. Got it? Now if at all you consider a stop codon, so you have to minus 1. But here you have clearly saying that stop codon has not been mentioned. So your answer is this. Cool. Next, statement number 1. Deoxyribonucleotide triphosphate acts as substrate during DNA replication. What do you mean by that? So basically here is your DNA strand. Okay, A, G, C, T, and the list goes on. So when I say that I am bringing nucleotides for replication process to happen, for a new DNA strand to be made, you need to understand that these nucleotides are in the form of DNTPs. What do you mean by DNTPs? DNTPs means you have the deoxyribose sugar over here, the nitrogenous base attached over here. Okay, so it can be, suppose it is adenine over here, then it is thymine over on this particular side. Yeah, and in the fifth carbon, my dear children, there is not just one phosphate group. This whole thing is a nucleotide, I agree. But please remember, it is actually triphosphate that comes. Now next time another nucleotide comes over here, my dear children, the third carbon of this and this particular phosphate will be making a bond, breaking this particular bond out there, breaking this particular bond. And it produces high amount of energy which allows joining of this particular point. You get it? So it is DNTPs that comes over here. DNTPs are basically the substrates and they are also the energy source. Why energy source? Because when the phosphate groups bond breakdown over here, it provides the energy for the formation of the next bond. Okay, so deoxyribonucleotide triphosphate DNTPs acts as substrate during DNA replication. Correct. And statement number 2, deoxyribonucleic triphosphate also acts as source of energy in the process of DNA replication. Correct. It's both substrate and energy source. So I'll say both statement 1 and 2 are correct. Got it? Yes. Fine. Now, and that is how basically nucleotide chain is being formed every single time. Okay. In a pea plant, each character is governed by a gene which is pea plant. Monocystron, polycystron. When the term comes in the question, just understand you have to think about prokaryotes and eukaryotes. Okay, polycystron, P for polycystron and P for prokaryotes, as in you're talking about bacteria and all the things. Monocystonic, that means you're talking about eukaryotes out there. What do you mean by monocystron? Basically, this cystron is only going to have one gene, and this one gene codes for only one protein. This is known as monocystonic nature of the genes. In the case of bacteria, you have polycystrons. That means they will have multiple gene segments, and these gene segments, my dear children, will be producing multiple proteins as well. That is what you mean by polycystonic nature. Okay, that is found in prokaryotes, monocystonic in eukaryotes. Now in pea plant is what? Pea is a plant and plants are eukaryotes. Anything apart from bacteria is what? A eukaryote. So just understand that anything apart from bacteria will be monocystonic in nature. Okay, yes, fine. Next thing, which of these are correct? UTR are located on the DNA, untranslated regions. Translation happens where? In mRNA. So do you think untranslated regions is there on the DNA? No, Baba. Is there in the mRNA. UTR is present both at the 5' end, before the start codon, is it? Yes, it is present. And also at the 3' end, after the stop codon. Is it? Yes, correct. UTR provides stability to the mRNA. Does it provide stability to the mRNA? Correct. UTR is required for efficient translation. Do you think you require UTR for efficient translation? Again, correct. You require it for efficient translation. So 2, 3 and 4 is correct answer. 2, 3 and 4, that means D option is correct. Remember, these are the functions of UTR. It helps in initiation, it helps in exit also, that means termination also. It is present at the both 5' end before the start codon and after the stop codon at the 3' end. Please remember, where is this 5' end start codon, 3' end stop codon? That is also important. Sometimes they can give you that and get you confused. UTR provides stability to the mRNA. Yes, it does. So yes, your answer for this question is D. Cool? Cool. Next, in a DNA strand, if there are 100 nucleotides and 30 are adenine. In a DNA strand, if I am saying that 100 nucleotides are there, out of that 30 of them is adenine, okay? And the number of thymine, guanine, cytosine are, okay. So we just have one data and we have to find what is this number, what is this number, and what is this number. How can we find that? Remember, how many, this is DNA strand, as in double-stranded DNA is what you're talking about over here. How many ever adenine is there, that many thymine is also going to be there, because A base pairs with T, right? So here also it will be 30. Now remember, you are given that 100 of them is there. In total you have 100. Out of 100, 60 of them are adenine and thymine. How 60? 30 plus 30 is equal to 60. So what you're left with? You're left with 40. Now this 40 you have to divide between both of them, because this, if this has 20, this will also have 20. 20 plus 20 is equals to 40, and you have got the answer. Thymine is going to be 30. You please know the order. Thymine is 30, guanine, my dear children, is going to be 20, and cytosine is also going to be 20. So your answer for this question is going to be A. It is going to be A. Got it? Yes. Yes. And that's how we finish that particular chapter also. Again, my dear children, there are so many other things also. We will be seeing that it's not like everything I can complete over here. In 2-3 marker questions, in 5 marker questions, we will be seeing more concept. So make sure you watch that video, okay? Yes, and then don't tell me, ma'am, how it is possible. So yeah. Okay, now we are going ahead with the next chapter, that is Principles of Inheritance and Variation. If at all you have reached up over here, type you have crossed, level 2 crossed. Okay, level 2 crossed. Okay, so let's get into this world. So question number 1, progenies with A, B and O blood groups are not possible from which of the following mating parents? You have been asked that A, B and O blood groups should not be produced, are not possible from which of the following parents. So for A, B to come, you understand, for A, B to come, one parent should have at least one gene, that is A, and the other parent should have another gene, that is B. Right. For O blood group to come, my dear children, one parent should have O and other parent should also have O. Then only O blood group will be coming in. Now I have to make sure that basically O ka I cannot guarantee, okay, but for this I can take the help of the information given over here. If I have A on this side and B on other side, that means there are chances of AB blood group to be formed. So here you have been asked which one progenies with AB and are not possible from which of the following parents. So you have to exclude the ones from which it is possible, right? So from here it is possible because A is here, B is here. From here also it is possible because A is here, B is here, A is here, B is here. So wrong. A is here, A is here, fine. A is here, B is here, opposite side, so again wrong. So basically, my dear children, you are just left with this particular option. Here, if the parent is O blood group, that means both this are OO. Here the parent is AB blood group, that means it is like this. This and this fuses, you get IA, IO. That means it is going to be A blood group. This and this fuses, you get IB, IO. That means it is going to be B blood group. Why? Because B is dominant over O. This and this fuses, again IA, IO. A is dominant, A will be shown up. When this and this fuses, again you will get B blood group. But anyway, AB and O is not there, which is why your answer for this question is going to be A. Got it? Yes, got it. Fine. Next question. What is the possible blood group of an offspring when both the parents have AB blood group? Now you tell me the answer. If both the parents are having AB blood group, my dear children, A, B, B, A and B, okay, this and this comes together, you get IA, the blood group is A blood group. This and this comes together, you get IA, IB, the blood group is going to be AB. Both of them are co-dominant over here. This and this, again IA, IB, again showing co-dominancy AB. This and this comes together, it is IB, that means B blood group. So how many possible things are there? Oh, okay, just a second, okay. What is a possible blood group of an offspring when both the parents have AB blood group? It will be AB, A and B. AB, A and B. So your answer for this question is B. This is the answer. Okay? Yes. Fine. Now, genes located very close to one another on the same chromosome tend to be transmitted together and are known as genes which are very close to each other, like this. They are known to be basically going to be transmitted together. Why? Because on the chromosome they are very close to each other. Even if there is cutting and splicing and things like that happening, my dear children, what is there? These two genes are always going to be close. What do you call them as? These are known as linked genes. And when they are very close to each other, you call them as tightly linked genes. Tightly linked genes. And one very important information from Jasmine Baba, and that is basically tightly linked genes will have lesser recombination. That means they will not be forming new combinations. They are very close to each other. There is no point of formation of new combinations. So I'll say lesser recombination. Remember that. And if they are loosely linked, they are far away from each other, they will have more recombination. Okay? Okay. Next, try it out. So I have given a question for you to try it out. Which three scientists independently rediscovered Mendel's work? Any idea? So Mendel was very sad because his works were not recognized. Later on, after 37 years, my dear children, his works got recognized. By whom all? So please let me know in the comment section below. You have got the question. You need to give me the answer. It is given in the NCERT. Just before chromosomal theory of inheritance, you will get the answer. If at all you do not know. Yes. Now, a plant having the genotypes capital A small a, capital B small b will produce. So you can give me answer like this, homework. Okay. So plants having a genotype capital A small a, capital B small b will produce dash kinds of gametes. How do I know how many type of gametes are going to be produced? So the question is very, very simple. If at all you know the formula goes like this. 2 raised to the power n. N is the number of heterozygotes. The number of heterozygotes. And what is it? Any idea? This is a heterozygote because here it is capital A small a. Both of them are different alleles, so I'll say this is heterozygote. So number 1 heterozygote. This is also heterozygote, number 2 heterozygote. So I'll write 2 raised to the power 2. Right? 2 raised to the power is equals to what? Is equals to 4. So 4 type of gametes are going to be formed. Your answer for this question is B4. Okay? Okay. Fine. Now the next question that we have, which of the following is not a pair of opposing characteristics Mendel studied? Which of the following is not a pair of opposing characteristics that Mendel studied? So Mendel has 7 pairs of contrasting characters that he studied. Okay, which one is not? Axil and terminal flowers, this he studied. Which one is dominant? Axil one is dominant. Axil means over here from the position where you have buds and all, you have the flowers. Terminal means at the tip. So which one is dominant? This one is dominant. Okay, full and constricted pods. Full means nice pod, okay, which has the green peas in that is full. Constricted means it is like this. Which one is dominant? Full one is dominant. Green and yellow pods. So when you go to the market, which one do you pick? Green color ones or yellow ones? It will be green. So that is dominant. This is also correct. Pink and white flowers. Mendel, if he was here, he would have given me the answer and said that, Papa, what question is given over here? It is purple or basically violet and white flowers, not pink and white. Okay, it is violet. So this one is the odd one out. Okay, and over here, these are the features that are given. Please remember there is a formula gravity for remembering all the dominant traits. G for gravity, G stands for green. So in the case of seed color, in the case of pod color, green is dominant, right? In the case of seed shape, R is dominant. So gravity R. So so I'll write it. I think that will be better. So in seed, we think that green is dominant, but actually in seed it is yellow, which is dominant in color. Okay, so please know that. In pod it is green. Yes. So G stands for green. R stands for round seed. A stands for axil flowers, V stands for violet flowers, and I stand for inflated pods, or what do you call as full pods. T stands for tall, and Y stands for yellow seed. Okay, so gravity is basically for all the dominant traits that you need to remember. Cool. Now, chromosome maps or genetic maps were first prepared by. Chromosome maps. So basically, it's like on the chromosome at this position, this gene is located. Who discovered that? It was Sturtevant. Sturtevant is what? Alfred Sturtevant is a student of Thomas Hunt Morgan. Thomas Hunt Morgan gave the idea about linkage and recombination. He said that genes basically which are very close to each other, they show lesser recombination. So with the percentage of recombination, if at all the percentage of recombination is more, that means they are far from each other. And if that is understood, basically I can locate the genes also. So basically, if I'm saying the recombination percentage is more, that means the genes are very far from each other. And if I have the data for recombination percentage, with that I can also understand where are the genes located as well. Okay, so the work was done by Thomas Hunt Morgan. He gave down two things, that is this, as I said, linkage and recombination. Sturtevant went ahead and gave an idea about the actual thing, that is genetic map. Okay, or gene map. Yes. Next thing, which of the following is incorrect regarding ZWZZ type of sex determination? ZWZZ type of sex determination, if at all you remember, my dear children, ZWW stands for woman. Over here is the female who are going to produce two type of gametes, one Z and another W. Usually in our case, in the case of human beings, it is XXXY. XY is produced by males. Males produce two type of gametes. But here, females are the ones producing two type of gametes. This is known as female heterogamety. Heterogamety means different type of gametes. And it is only seen in the case of birds. In the case of birds. So it occurs in birds, correct. Females are homogametic. Arre baba, they are not homogametic. Males are homogametic over here. Females are heterogametic and males are homogametic. Females are heterogametic, males are homogametic, correct. How can there be? Okay, you have to find what is the incorrect one. Yeah. Okay, just a second. Yes. Total number of chromosomes is same in both male and female. Yes, it is. It is very much same in both male as well as female. So this is also correct. What is wrong over here? Only one option is wrong over here, and that is this one. Okay, just a second. So when I mark things wrong, I put the answer like this. This is the answer. Female or heterogamety, that is correct. Okay, this one is the wrong one. Cool? Yes. Okay. Can we go ahead with the next question? Please know all the different types of sex determination. In honeybees, the number of chromosomes are not same in the case of male and female. In the female, there are 32 chromosomes. In the case of male, there is only 16 number of chromosomes. Okay? Yes. Half the number of chromosomes. Okay. Now let's go ahead. A woman with normal vision whose brother is colorblind. Okay. Now this is colorblindness related thing, so you have to be very, very careful. A woman with normal vision whose brother is colorblind marries a man with normal vision. They have 4 children, 2 sons and 2 daughters. One son is colorblind while the other son and both the daughters have normal vision. The pattern of inheritance for the diseases. So over here, it's such a simple question. Listen to me carefully. They just put all this drama to confuse you guys. First of all, to scare off you guys. Here they have clearly mentioned color blindness. So you know color blindness is a disease. What is color blindness? It is sex-linked recessive disorder. We all know that color blindness and hemophilia. Color blindness and hemophilia, both of them are sex-linked recessive disorders. So can I just write sex-linked recessive? Do I need to even think for a second? Okay, so your answer for this question is going to be A. Fine, ma'am. Okay. Now the next thing, if both the parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? So they have mentioned it is autosomal recessive disorder. That means it is not going to affect any sex chromosome, it is affecting your autosomes. In your autosome, some mutation is there, that is why this disease is there. And they have also mentioned that both the parents are what? Unaffected carriers of thalassemia. Okay. So in thalassemia, your hemoglobin is affected. So I'll just write like this, okay? So suppose this is a normal one and this is the affected gene. This is a normal one, this is the affected gene. They are the carriers. The carriers will have one affected and one normal. The normal will be dominant over this, which is why they will be just a carrier, they will not have the disease, right? So you have been asked how many of them are going to be affected in the children. That is the question. So let's do the calculation. This and this goes. HBA is going to be a normal child. This and this, it will be HBA and HBT. they will be a carrier child. We do not know he or she because it is autosome linked. It is not sex chromosome linked, right? Now this and this, HBA, HBT. Over here, again, my dear children, they are going to be carriers only. Lastly, you have HBT and HBT. This one, both the genes are being affected. Okay, both the chromosomes are being affected, which is why, my dear children, there is no chance that a normal baby can be produced over here. So I'll say out of 1, 2, 3, 4, one child can be affected. Out of 4 children, one can be affected. That means, can I say 25%? Yes, I can say that. You get it? So please know, whenever you're speaking about autosomal recessive, there's no male and female over here. All of them can be equally affected. We do not know the sex of the child. You just have to look for the percentage of the ones which are affected. Okay? And they will be carriers. They're going to be carriers. And carriers are not counted as affected. So yes, if they're asking you about carriers, please mention about that as well. So yeah, these were the question. Again, as I said, so which ones were difficult? I have taken that. Apart from that, if at all there is a concept which is super tough, I might have explained it 2-marker, 3-marker sessions. So please watch that. As I said, each and every part of the NCERT over here cannot be covered. It has to be distributed in different range of marks, right? So please have that in your head and then only go ahead. Yeah, these are the most probable MCQs that I'm sharing with you. The tougher ones, some of them might be easier also, but yes. Okay, cool? Yes. Now, human health and disease is the next chapter. Over here, I have the first question that is up there. Human immunodeficiency virus replicates in. So I think we have to be quick because it's already 1 hour 50 minutes, right? So let's go. Human immunodeficiency virus replicates in B cells, NK cells, helper T cells and PMNL. What do you think is your answer? HIV replicates in actually macrophages. First of all. Then later it is going to affect the helper T lymphocytes. This is the major target, but first it goes and infects the macrophage and increases the number. That is why macrophage is known as HIV factory of the cell. And then later it affects the helper T cells, which is the major target as well. But anyway, since macrophage is not been mentioned, you have to go with this. Not B cells, not natural killer cells. PMNL stands for polymorphonucleoleucocyte. You also call them as neutrophils. That is not the answer for this question. Okay. Next thing, the drug which is depressant and slows down the body function is. It is a depressant, okay? It slows down your body function as well. You just have one drug such like that, and that is known as heroin. Okay, it is known as heroin. And do you see heroin in these options? No, that means other name of heroin is given. What is the other name of heroin? Diacetylmorphin. Because you have morphine and heroin in one particular category. When you do acetylation of morphine, that's how you get what do you call heroin. Okay, so it is known as diacetylmorphin. If that name is also not given, you have to go for the other name, that is SMAC. That is SMAC. Okay, so heroin, SMAC, diacetylmorphin, all of it is same. Bitter, odorless, colorless, a crystalline substance which is there. Okay, it can be taken using snorting or injection. Yeah? Yes. So please know about that. Next one, and you get it from the latex of the poppy plant. So morphine, I have written diacetylmorphin, or you also call this SMAC. Next thing, symptoms of constipation, abdominal pain and cramps associated with the disease were what? So there is constipation, okay, because of which you have abdominal pain, and there is cramps also. This is seen in which of the diseases? This is your MQP1 question. Amoebiasis, richness, nostoc, ovulation, colostrum and diversity. Just fill in the blanks question. The answer is amoebiasis. And who is the causative agent? It is Entamoeba histolytica. It causes amoebic dysentery, or you also call this as amoebiasis. Either way, that is your answer. Okay, the next question that we have is question number 4. Antivenom given against snake poison contains what? Antigens, antigen-antibody complex, antibodies or enzymes. What do you think is the answer for this? Antivenom which you give against snake poison. Suppose a snake came and attacked you. Okay, now the doctor will give antivenom. Why? Why antivenom? Because at that particular point of time, basically the body needs ready-made antibodies. Okay, you cannot wait for your body to produce antibody by itself. Please understand that. You need ready-made antibodies, and that is what you get from anti-venom or basically antitoxin that is given in the case of tetanus injection also. Okay, so please know, it is not antigens, it is not antigen antibody complex, it is just antibodies that is being given. Okay, and not even enzymes. Yeah. Next one, the substrate produced by a cell in viral infection that can protect the other cells from further infection is. So there is a substance that is produced from the cell and it is protecting the other cell from infection. So this cell has been infected from virus. What is it releasing? So that it protects the other cell, it is going to be cytokines, and the name of the cytokine is interferon. Cytokine means some secretions of the cell, okay, and here you have a specific name for that. It is interferon. Always remember, interferon is a famous guy among the virus infected cells. Now there is one more interferon being mentioned in NCERT, and that is alpha interferons. Alpha interferons are given to cancer patients because it is a biological immune response modifier. It's something that is boosting up your immune system. That is the case of cancer, and that is alpha interferons. This is plain interferons, okay? Please remember that. Okay. Now, DASH is a very effective sedative and a painkiller. Sedative. So there are two drugs, as I told you, which is responsible for depression and all, depressant and slowing down of your body. Okay, one is morphine. It is actually sedative. It makes you sleep, and also painkiller, which is why doctors use it on patients who have underwent very bad surgeries. Okay, and after that, as I said, from morphine you get heroin, which is depressant, slows down your body as well. Here you have been asked about sedative. That means this particular drug makes all people sleep. Okay, sedation, it gives sedation. And also it's a painkiller, it has to be morphine only. Okay, and both of them are opioids. Yes. Now, which of the following pairs correctly matches a disease and a pathogen causing it? Typhoid is caused by Salmonella typhi. Is it? Yes, definitely. This is the first one itself is the correct one. Let's see why others are not. Pneumonia is caused by Haemophilus pneumoniae. Oh, Haemophilus pneumoniae. This also is correct, right? No. Haemophilus pneumoniae is not the correct one. It is Haemophilus influenzae. It is Streptococcus pneumonia. Two bacteria causes pneumonia, right? So that is why it's wrong. Malaria is caused by Ascaris lumbricoides. Papa, Ascaris lumbricoides is like, I did not cause this. Do not blame me for this. Okay, so malaria is caused by Plasmodium, not by Ascaris lumbricoides. And it causes ascariasis, by the way. Ringworm is caused by Antamoeba histolytica. Okay. Is it? No. Ringworm is not caused by any worm also. It is caused by fungus, trichophyton, epidermophyton, microsporum and things like that, which is why this is also wrong. So your only answer for the question that you have is A. And by this you understand that all the diseases and the causative agents are important for you to know. Yes. Next, hemozoin is a dash, precursor of hemoglobin. Is it? Hemozoin, is it a precursor of hemoglobin? No. Hemozoin is basically what? Basically, the plasmodium which is there, it infects your RBC. Inside that, what do you have? Hemoglobin. This globin is what the plasmodium needs. Okay, but it breaks it down. You get heme plus globin. This heme which is produced is very, very toxic for the plasmodium, which is why it converts into hemozoin. The thing is, this hemozoin, when it is released from the cells, how it is released? Over here you have so many plasmodium also. When they flourish too much, it is going to burst open, and that's how hemozoin is going to be released. This hemozoin will be causing chills and fevers to us because it is toxic for us instead. You get it? So over here, it is precursor of hemoglobin, wrong. Toxin released from Streptococcus infected cells, Streptococcus, papa, it causes pneumonia, so wrong. Toxin released from Plasmodium infected cell, yes, exactly. Toxin released from Haemophilus infected cells, no. So your only answer for this question is going to be C. And over here, I have explained that again in the life cycle of Plasmodium. I will be talking everything in detail. Okay, so it enters, Plasmodium enters in our body in sporozoite stage, exits from our body in gametocyte stage. No sexual reproduction happens in our body. Okay, sexual reproduction happens in the mosquito's body. Okay, so please remember all those things. Sporozoite is the case where it enters through saliva, and basically it exists in the gametocyte stage. No sexual reproduction happens in human body. Only asexual reproduction happens in the human body. It affects your liver first, then your RBCs. Cool, cool. Yes, fine. Now, Next question, which of the following is an autoimmune disease? Autoimmune disease means your cells do not know that, okay, these are also my cells, I should not be killing them. So that is autoimmune disorders, okay? Whenever your cells, your body is not able to recognize what is self and what is not self and keep on killing your own cells also, that leads to autoimmune disorders. And over here in your NCERT, you just have one example, and that is rheumatoid arthritis. Rheumatoid arthritis is the only example for autoimmune disease in your NCERT. Yeah? Yeah, cool. Next question. The site where lymphocytes interact with antigens and proliferate to become effector cells are. The site where lymphocytes interact with antigens and proliferate to become effector cells. Effector cells means actually do their function of killing the antigens. So whenever lymphocytes are going to kill the antigens, that is going to happen only in the secondary lymphoid organs, not in the primary lymphoid organs. In the primary lymphoid organs, they become antigen sensitive. Remember the term, antigen sensitive. They know that I have to kill an antigen and things like that, but they don't kill it. They kill only in the secondary lymphoid organs, and that is when we say that they have become what? Effector cells. So where do they become effector cells? You have to spot the secondary lymphoid organs. Thymus is a primary lymphoid organ, which is why it is wrong. Bone marrow and thymus, again wrong. Both of them are primary lymphoid organs. Peyer's patch and tonsils, both of them are secondary lymphoid organs, which way this could be the answer. Bone marrow and spleen, wrong. Listen, over here, lymph nodes, spleen, Peyer's patch, tonsils, all of them are secondary lymphoid organs. But here they are paired with primary lymphoid organs, which is why they are wrong. Only one option is correct, and that is C. Cool? Cool. Next, injection of antitoxin in tetanus confers which type of immunization? Listen, over here, antitoxin, I told you, you are giving ready-made antibodies. You're not waiting for your body to produce antibodies. So this type of immunization is passive immunization. When your body produces antibodies by itself, you call it active immunization. That happens in vaccination. That happens when you get infected with something naturally. Your body produces antibodies, right? That is active immunization. Over here, you are directly giving ready-made antibodies, which is why it is passive form of immunization. So your answer for this question is going to be B. Cool? Cool, ma'am. Okay. Tobacco consumption is known to stimulate secretion of adrenaline and noradrenaline. The component causing this could be, in tobacco, what is that something that increases your blood pressure? It is nicotine. Nicotine, my dear children, is responsible for basically stimulating the secretion of adrenaline and noradrenaline. Now, when these hormones are secreted, your blood pressure is going to go high. Always remember, if there's a question coming in, which is the drug responsible for increase of blood pressure? It is nicotine, and you get it from tobacco consumption. Okay? Okay. Next one, charas and ganja are the drugs which affect. Charas, ganja, hashish, marijuana, la 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 la. Everything you get from cannabinoids, and cannabinoids affect your cardiovascular system. The only ones which are affecting cardiovascular system. Rest, opioids as well as coca alkaloids, both of them affect the CNS, okay, but not the cardiovascular system. So this guy affects the cardiovascular system. These are cannabinoids. They are cannabinoids. Okay. Next one, a chemical structure is given below. Identify its plant source. So it looks like an O, which is why I'll say it's opioid. And in opioid, you have two, morphine as well as heroin. Which one is this? It is actually morphine. And you get it from Papaver somniferum. You get it from Papaver somniferum. So basically, they're not even asking you what is the structure. They're asking you from where do you get it. So high level of things, you need to an understanding of that, right? So C. Fine. Next, the disease chikungunya is transmitted by. How do you think chikungunya is going to be transmitted? Okay. It is what? Sometimes I forget that I'm a teacher, and sometimes, most of the times, guys, first of all, let me tell you, I'm your teacher, but I'm also your friend. So yes, please consider me like that also. So your answer for this question is going to be Aedes mosquito. Okay, there's going to be Aedes mosquito. It is the one that causes chikungunya as well as dengue. You have Anopheles mosquito that causes malaria, and you have Culex mosquito that causes filariasis. Okay, so B is your answer for this question. Cool. Next one. Identify the enzymes which deviates from the central dogma of genetics during HIV infection. Okay, what is the central dogma of genetics? The DNA is going to form the RNA. The RNA is going to help in the formation of protein, and DNA forms itself. That is known as replication. This is central dogma. Now you have been asked, there is an enzyme which deviates from this. that deviates from this. What is that enzyme? It is reverse transcriptase. Why? Because in the case of HIV infection, the RNA of the HIV virus reverse transcribes to form the DNA, and that has been done using the enzyme reverse transcriptase, because our cell has DNA and it wants to incorporate it into our DNA. That is why it does reverse transcription, and that is opposite of the central dogma which was given by Watson. Okay? Yes. Finally, now we get ahead into the next chapter, that is evolution. So in evolution, we are going to deal with the MCQs, and by the way, I forgot how many levels have been crossed. So do you remember the last level I remember was in principles of inheritance and variation. It was level 2 crossed, right? So now let's go ahead and cross level number, where is that? Okay. Level 3 crossed. If at all you have crossed level number 3, let me know in the comment section. By the way, was it level number 3? Okay, I remember one more level number 4, but anyways, okay. Starting with the first question, select the correct match. Australian marsupials is given on this side, placental mammals are given on this side. So spotted cuscus is from team Australian marsupial. You have to find the opposite of it from here. What is the answer? Flying squirrel, bobcat, anteater, lemur. The answer is lemur. Ma'am, how do you remember it? For flying squirrel, the opposite one that you have is flying phalanger. For bobcat, you have Tasmanian cat. For anteater, you have numbat. Okay, so how do you remember all of that? Once you go through this, slowly you will be remembering all of it. Remember, placental mammals are the ones with smaller names. Australian marsupials are the ones with larger names. So if mole is there, marsupial mole is there. Okay. If anteater is there, numbat is there. This is something different. So you need to remember anteater and numbat. Mouse is there, marsupial mouse is there. Easy. Lemur is there, spotted cuscus is there. This is also different. So please remember it. Flying squirrel, flying phalanger is there. Okay, somehow different. Bobcat, Tasmanian tiger cat. Tiger cat is there, so it's easy. Wolf, Tasmanian wolf. So always remember placental mammals, smaller name. So placental mammals, Baba, smaller name will be there. Smaller name is going to be there. Here you have bigger names. So mole, marsupial mole. Which one is placental mammal? This side is placental mammal. Okay. Anteater, numbat. So one which I have colored have different names, which is why you need to remember them specifically. Anteater, numbat, lemur, spotted cuscus, flying squirrel, flying phalanger. Phalanger comes that side. Okay. In Australian marsupials it comes. In Australian marsupials it will be coming. Please remember. Okay. Yes. Okay. So I was thinking of a way for you to remember it, but apparently flying squirrel, no. Anyway, phalanger is a bigger name, so you can remember it comes from the Australian marsupial side only. Fine. Next question. Which of the ancient men protected their body with hide and buried their dead? Okay. So, you got Homo erectus. Okay. 1.5 million years ago, you got Homo erectus, which started hunting food as in meat. They have two things, the divergent of two things. One is Neanderthal man and one is Homo sapiens, that is us. First who came? Neanderthal man. And they were the ones with 1400 cc of brain capacity or cranial capacity, and they started hiding their body and also burying their dead. So it is Neanderthal man, which is the answer for this particular question. They are very unique for that particular reason. Cool? Cool. Next thing. Okay. So I've explained everything. Again, you can go read the PDF, or I will be explaining you in 2 marker, 3 marker, do not be worried. Okay? Next, arrange the following animal groups in current sequence of their occurrence on geological time scale. Okay, amphibians, fishes, reptiles, mammals. Very easy way to remember. Farm. First came fishes, then came amphibians. From amphibians you got reptiles. From reptiles you got both aves, that is birds, and also mammals. So you have to go with farm. Farm basically means second comes first. Second. So these two options are wrong. Okay, after second you have amphibians, 1. So this is your answer. 2, 1, 3, 4. 2, 1, 3, 4. Okay? Okay. Yes. Next one. The paleontological evidence for evolution is obtained from paleontological, as in fossils. Okay, you get the paleontological evidence of evolution from the fossils. So your answer for this question is going to be fossils. Easy question. Yeah? Yes. Next thing. Observe the following diagrammatic representation of operation of natural selection on different traits here. Natural selection depicts what? Stabilization, directional, disruptive cannot be depicted. Listen, over here you see two bumps, right? So listen to me carefully. If at all you have tall and you have dwarf, and if both the characters are being selected, you say that this is disruptive. Two extremes are being selected. If at all we say the median character is being selected, that is middle height, then it is going to be stabilizing. If at all we say that only one, dwarf ones are selected, then I say it is directional. Or tall ones are only selected, then I say it is directional. So this is disruptive where both the extremes are selected, stabilizing as in basically the middle character is going to be selected, and directional as in basically one extreme is going to be selected. So here it is going to be disruptive. C is your answer, it is disruptive in nature. Cool? Cool. Fine. The next question. Dash stage in human evolution used heights to protect their body and buried their dead. So what is that stage that uses heights to protect their body and bury their dead? It is going to be Neanderthal man. We just discussed about it. Okay? Yes. Statement number 1. Man has domesticated dogs and has created different breeds by artificial breeding. Is it? Yes. It is definitely true. That is how we got different species of dogs out there. Statement number 2. Intensive breeding of dogs signifies divergent evolution. Is it? What do you mean by divergent evolution? Means you had a common ancestor. Now basically they have diverged into different species out there. So is it like that? Yes. Yeah, because so husky you find it in colder areas. Okay, other type of dogs you find it in different. Indian dogs are different from how the other dogs are being seen. You get it? Why? Because they are in different habitats. So obviously they have diverged, not converged. They have diverged. So can I say both statement 1 and 2 are correct? Yes, it won't be wrong to say that both statement 1 and 2 are correct. Next question. Statement number 1. Antibiotic or drug resistance is due to evolution by anthropogenic actions. Okay, what do you mean by anthropogenic actions? It means man-made actions. So if I say that we are resistant to antibiotics or drugs, do you think it's because of our actions? Yes. Why? Basically we are giving high doses of medicine and we are taking it very usually, such that the organisms which are there, they're mutating, and they're mutating in a way that they are okay with all these changes out there. Okay, they are being resistant to that. So yes, it is an example of evolution by anthropogenic action. That is absolutely true. Similarly, plants becoming resistant to pesticides, or basically not plants, the pest becoming resistant to pesticides, that is also an example for anthropogenic action. Now statement number 2. Many antibiotic or drug resistant organisms arise due to overusage of antibiotics. Yes, because they thrive, they want to survive, which is why they mutate, and then the one that is able to, it is just surviving and keeps on reproducing. So yes, I'll say both the statements again are going to be correct. Cool? Cool, ma'am. Okay. A brown-billed duck is newly added to a farm of white-billed duck. This indicates what? There were a brown-billed duck and now soon I am adding some white ones here. Can I say it's gene flow or gene migration? From some other population, I'm adding something over here which is going to change the frequency of the alleles over here. Isn't it? This is an example of gene flow. If at all I was doing, it was happening by chance. Suppose the black ones or the brown ones will be killed by some attack and accidentally all the brown ones lost their life and the other ones were living. That means it is genetic drift. But here it is gene flow. Why? From outside I am bringing it inside, and that is changing the genetic frequency, which is why it is known as gene flow or gene migration. Suppose there are white people in America, you go there. Okay, we are blacks, okay. That does not mean I'm racist, okay, please understand that. I'm giving you an example, and by we I mean all of us, okay, not you, it's we. So when we get inside, there's a change in the genetic frequency. Similarly, when they come over here, there's a change in genetic frequency out there here as well. That is gene flow or gene migration. Okay, okay, fine. Now change in frequency of genes and alleles is due to, change in frequency of genes and alleles is due to mutation. Yes, mutation can bring in changes. For example, I had the brown ones and the black ones. Now because of mutations, I have blue ones also. So yes, it is changing the frequency of the alleles, correct? Gene flow, we just saw an example for that. Yes, that is also a reason. So can I say both A and B is the answer? Neither A and B cannot be true. So both A and B can be true, which is why I'll say C is the answer for this particular question. Works? Works. Definitely works. Next, the first amphibians that lived both on land and the water evolved from. The first amphibians that lived both on land and the water evolved from, listen, you had fishes which was living in water, you had amphibians which lived on both land and water. So between fishes and amphibians, you had one organism that is known to be the connecting link, and that is lobe fin. By that I mean, this was just like a fish, but the fin which was there, it was very hard, such that with the fin it could walk around. And that is the connecting link between the modern day frogs and salamanders and also the fishes. So I'll say lobe fins, or they can just give you coelacanth. Coelacanth. Please know that this name is same only. They are used interchangeably. Okay. Similarly, between reptiles and mammals. Reptile and mammals, you have connected link which is known as shrews. Okay. Yes. Next, the biggest land reptiles that existed during Mesozoic era was. Mesozoic era is made up of Triassic, Jurassic and Cretaceous period. Okay. So over here, my dear children, the biggest land reptile that existed was Tyrannosaurus rex. Tyrannosaurus rex. That is going to be your answer. B. Okay. Next, statement number 1. Hugo de Vries believed mutation leads to spontaneous evolution. Hugo de Vries said that mutations are happening, which is why, and these mutations are really fast, it happens, and that is how new evolution is taking place. Yes, he was correct when he said that. Now, mutations are random, directionless, causes saltation. Mutation, do you think it is planned? No, it is very random thing. Yeah, does it have any direction? Does it support anything? No, it is directionless. It can happen in any direction. Okay, anything can be selected out there. Does it cause saltation? Saltation means one large mutation can lead to formation of a new species altogether. One step mutation can lead to formation of a new species altogether. Do you think that can happen? Yes, that can also happen. So can I say both the statement 1 and 2 are correct? Yes. So according to Darwin, he said that there are some changes happening that slowly leads to gradual changes out there, and that leads to evolution. Okay, so Darwin was promoting gradual changes. He was talking about mutations, which is very, very fast. Both of them were ideally correct in their own places. Both of them spoke about evolution and both of them were correct. So yes, okay, but Darwin spoke about gradual ones, he spoke about spontaneous ones. Cool. Now, elongation of neck in giraffe due to inheritance of acquired character was given by. So basically, there was a guy whose name was Lamarck. He said that whenever you use an organ, basically because of its use, it stays there. Okay, and if at all it is not being used, basically it's not there. So there were actually smaller giraffe, but the smaller giraffe, they were using their neck to go up and take up the leaves out there, and since they were using, that is how their neck got elongated. But to contradict that, Darwin came and said that how can that be true? Acquired characters, something that you acquire during your lifetime, cannot be passed to the next generation. That cannot happen at all. Okay, prove it then. So basically, experiment was done with mouse. The tail of the mouse was being cut, and it was seen that, let's see, the babies of the mouse, will it have tail or not? Because anyway, it does not have tail, right? So let's see it if the babies have. The babies had tail. That means the acquired character of not having tail is not being passed down, right? Similarly over here, it's not like the giraffe is not utilizing its neck. Probably some mutations might have happened. Some might have shorter necks, some might have longer necks. The longer ones were able to survive, which is why they kept on reproducing, is what Darwin said. You get it? So the opposite was said by Lamarck, and please remember, it was being removed also. It was like, no, it does not make any sense. Cool? Cool, ma'am. Okay. Next, single step large mutation leading to speciation is also called as. Single step large mutation that leads to new species formation is known as what? Natural selection, branching descent, founder's effect, saltation. Answer is saltation. This comes below Hugo de Vries. Single step large mutation leading to speciation is known as saltation. Very, very important. Cool, cool. Next, the organic compounds found in the Miller's experiment are. Always remember, in Miller's experiment, he was only able to form one organic component, and that was amino acid. Nothing else. Rest of the experiments were done by different people, and they came up with different things. But Miller's experiment, Stanley Miller's experiment, he could only find amino acids. So your only answer is one only. Okay, one only. Nitrogenous base, pigments, fats and all were found by different other people out there, not by Stanley Miller. Cool. Embryological evidence for evolution was disapproved by. So embryological evidence said that basically fishes, amphibians, reptiles and mammals. That is how it goes, right? So since in the embryos of the mammals you have gill slits and you have tail also, but which usually goes in the adult state, it was said that whatever organism we have evolved from, basically the features of those organisms are seen in our embryo. So probably we might have evolved from fishes. That is what Ernst Haeckel said. Okay, that was embryological evidence. But then Carl Ernst von Baer, both of them are Ernst only, but the one with the bigger name was the person who disapproved this and said that that is not true, bro, because in that case amphibians features should also be seen in the case of mammals, reptiles features should also be seen. Why are we seeing only just gill slits which are there in the case of fishes, or just tails which are there in the case of fishes? That is absolutely wrong. If we have evolved from all these organisms, we should be having some characters of all these organisms. Why is not that like that, right? So that is how it was disapproved, and it was Carl Ernst von Baer, who was responsible for disapproval of this. Okay. Arrange the following plants as appeared on the different geological periods. So over here you have conifers, nitales, dicots and monocots. Listen, in the case of angiosperms, dicots came first and then came monocots. So this flow is okay. Here also the flow is okay. Here the flow is not okay. Here also the flow is not okay. So I'll just cut it off. Okay. Now what do we have? Conifers, nitales. So listen, conifers came first, then came nitales, then came dicots, then came monocots. So here nitales is given and conifers is given at last. That cannot be true at all. Conifers has to be coming front, then nitales. So if you see that, this is going to be your answer. Listen, look at this particular picture. Ferns, spinopsids. Basically, you are pteridophytes. Then you have conifers, ginkgos, cycads, nitales. So conifers came first, then nitales, then dicots, then monocots. That is how it usually grows. Okay? Yes, so that is your answer. Now 19. Replacement of the lighter colored variety of peppered moth, Biston betuloria, to its darker variety in England is an example of. So you have seen industrial melanism. You have seen industrial melanism where what is happening? The white ones were selected first because there was white background, there was lichen. Okay, later on when pollution was there, lichen is not there, white background is not there, which is why the white ones were spotted very easily. So it was nature which was selecting, it was not us, it was nature which was selecting, and that's all. So it is an example of industrial melanism, is an example of what? Natural selection. The one which is able to survive and reproduce gets ahead, right? So this is an example of natural selection. This is not regeneration, not genetic isolation, not even genetic drift. Cool, cool. Yes. Now at a particular locus, the frequency of allele A is the frequency of allele A. When you denote it by capital A, you just understand that this is the frequency of the dominant allele. So it is 0.6. Okay, and the frequency of allele small a is, that means you're talking about Q, it is 0.4. What would be the frequency of heterozygotes in random mating population at equilibrium? So how do you denote heterozygotes? Always remember there's a formula, P square plus 2PQ plus Q square. P square denotes the homozygous as in condition of capital A, capital A. So you have to do this. Okay, you have to do this. That's when you get homozygous AA. When you do homozygous small a, so that means you do 0.4 whole square, that's when you get AA. You have been asked about heterozygotes. That means A and A. How do you find that out? So P into Q. P is what? 0.6. Q is what? 0.4. Is that all? No. You have to multiply 2 also. So 0.6 into 0.4 is 0.24. Okay. Into 2 is equals to what? Is equals to 0.48. So your answer for this question is going to be 0.48. Okay. So do not forget to multiply the 2 over here. If at all you're asked about the percentage, write 48%. That's all. Cool? Yes. And now we enter the next chapter, that is Microbes in Human Welfare. One of my favorite chapters out there, and I see that 2 hours 23 minutes somehow as always has been done already. But are we going strong? I hope we are going strong, and we have reached the level 4. So level 4, if at all you have crossed, do let me know and let's go ahead. Saccharomyces cerevisiae, commonly called as brewer's yeast, is used for fermenting what? Malted what? Malted pulses, malted cereals, fruits, vegetables. Saccharomyces cerevisiae, which is known as brewer's yeast. In bakery industry, baker's yeast, okay? It is used for fermenting malted. Malted means something that has started with germination but process got stopped in between. That is malted condition, okay? So it was malted cereals that were being used. Malted cereals and also fruit juices are being used in this particular industry. Malted cereals and fruit juices, okay, in the brewery industry. So your answer for this question is B. This is this year's MQP question. Now, cyanobacteria is an autotrophic microbe widely distributed in aquatic and terrestrial habitats, can fix atmospheric nitrogen is cyanobacteria. In your NCERT, there is the name of 3 cyanobacteria that are given. One is nostoc, one is anabena, and the other one which is there, it is nostoc, anabena, oscillatoria. Okay, nostoc, anabena, oscillatoria. Okay, so instead of uno, you can put ano. That's all. So here nostoc is an option, so we'll go with that. If at all you're talking about bacteria, plain bacteria, there is rhizobium, there is azotobacter, and that is azospirillum. 3 things are also given. This is cyanobacteria, okay? They are autotrophic in nature. They can prepare their own foods. Normally, bacteria cannot prepare its own food. So here talking about own food preparing bacteria that can fix atmospheric nitrogen as well. That is ano. As I said, anabena, nostoc, and oscillatoria. Okay? They are used in paddy fields as well. Now, because of the presence of which of the following vitamin, curd is considered as nutritionally richer than milk. So, in curd, what do we have? We have vitamin B12, my dear children, which is why we consider it nutritionally more richer than milk. Okay, so when the lactobacillus or basically lactic acid bacteria is secreting lactic acid and converting or breaking down the casein protein which is there in the milk, it also adds up vitamin B12, and that is why the curd is rich in vitamin B12. Okay. Next, mycorrhiza is involved in absorption of dash from soil and passes it to the plant. What is mycorrhiza? Mycorrhiza is a combination of a fungus which belongs to glomus. And also the root of the plant. So basically the plant is going to be giving the product of photosynthesis, the organic food. Fungi, my dear children, provides phosphorus. Phosphorus, this is very, very important. Phosphorus, not nitrogen and potassium. It is phosphorus, okay, from the soil, because fungi can go deep in the soil and can trap it up. Apart from phosphorus, there are other things also that you get from this particular mycorrhiza. That is basically, it provides tolerance, it provides resistance, and it, increases the plant growth and development and all of these things. Okay, but here your only option that is given is phosphorus. Absorption of what? Phosphorus. Okay. Next thing, during sewage treatment, the gas produced in the anaerobic sludge digester includes. Listen, whenever the term anaerobic comes and whenever the term gas comes, just understand you're talking about biogas. Biogas is produced in anaerobic conditions by anaerobic bacteria. You call them as methanogens. Okay, and when you talk about biogas is made up of 3 very important things. One is methane, other one is hydrogen sulfide, and the third one is carbon dioxide. So your answer for this question is going to be C. So methane can be denoted like this. H2S is hydrogen sulfide, and carbon dioxide, you know how it looks. Sometimes instead of putting hydrogen sulfide, they can also put hydrogen. Please do not get confused. Your answer is going to be the same itself. Okay, done? Yes? Okay. Next question. Dash is a producer of butyric acid. Butyric acid is produced from a bacteria whose name is Clostridium butylicum. So if at all, again, you studied microbes in human welfare from me, I think some of you might be remembering the things that I have taught you, right? So Clostridium butylicum is the producer of butyric acid. It's a bacteria. Now, other name of the industrial chemicals are also given over here. You can just have a quick sneak peek, pause the video, and can just look at it. Now, Propionibacterium shermani is used in the production of Swiss cheese, okay, not in Roquefort cheese. In Roquefort cheese, you need a fungi. A fungi helps in the production of that. Swiss cheese is where basically big holes are there because CO2 is being expelled through that, okay, and it is being produced by Propionibacterium, okay, and it is Swiss cheese as the answer. So B is the answer for this particular question. Okay, I have mentioned it wrong. This is the answer, okay. So probably while copying and doing things like that from different there is a typist out there involved. There are a lot of things involved probably, which is why all these mistakes come. Do not be worried, I am here to correct them as well. Okay? Yes. Cool. Alexander Fleming discovered penicillin while working on. So Alexander Fleming was working on one very important thing that is known as, any idea? Any idea? Is it Streptococcus or Staphylococci? It is Staphylococci. He was working on Staphylococci. That's when he got to understand. He didn't wash the dish once and he thought that the bacteria is going to grow. But next day he saw that a fungal mold is growing out there, and because of this mold, the bacteria was not able to grow. Why? Because probably the mold was releasing something. What was it releasing? Antibiotic, that is penicillin. So the mold was known as Penicillium notatum, and basically it was releasing an antibiotic penicillin, and that did not allow Staphylococci to grow. So it is not Streptococcus. Streptococcus is responsible for production of, streptokinase, right? So remember that. This Staphylococci. And Alexander has L over here, Staphylococci also has an L. Okay, remember like that also. This is the guy. Yes. Now, which one of the following alcoholic drinks is produced without distillation? So, distillation basically means that I am making the water content to go away. When water is going away, the level of alcohol will be more concentrated, right? So, after drinking something, if at all it goes like this, that means it has underwent distillation in more amount. So, whiskey, rum, brandy undergoes distillation. You have been asked which is produced without distillation. That means water content is still there. It is not being removed too much. It is not too much concentrated alcohol out there. That means wine. Wine is something like that. Apart from wine, you also have beer. Cool, ma'am. Okay. Next, activated sludge is a sediment deposited in. Activated sludge. Think about it. So you had the primary effluent. You made it treat with one very important thing, that is aerobic bacteria and the fungal filaments together. It formed the flocks and things like that, right? It treated the organic matter out there. Now you transferred it. This was in the aeration tank. You transferred into settling tank. In the settling tank, the flock, my dear children, got settled down. And whatever is clean, that was sent into the river. This thing is known as activated sludge. The flock, basically, after the use, the flock which is there, it is the activated sludge. Where was it settling down? In the settling tank. So the first process is done in the settling. Primary treatment is done in the settling tank. Then for secondary treatment, you transfer it to aeration tank. Then back into settling tank. Then into anaerobic sludge digester. Okay, so first settling tank, then aeration tank, then again settling tank for this, and then you put this particular thing in anaerobic sludge digester. That is how it goes. Okay, so it settles down in the settling tank. A. Yes. Biogas is a mixture of, you tell me, biogas is a mixture of CH4CO2 CH2. Do you have H2S anywhere? No, you have hydrogen. Is that wrong? No, it's not wrong. So basically, this combination should be correct. Do not go and write CH3 out there. It is CH4. Okay? Okay, ma'am. Next. Match the column 1 with column 2 and select the correct option using the codes given below. Citric acid, cyclosporin A, statins, butyric acid. Okay, I'll just zoom this in and see. So citric acid is produced by Aspergillus niger. It is a fungus that produces it. Butyric acid by Clostridium botulicum. So A goes with 3. A goes with 3. A goes with 3. I have 3 options like that. I can just remove this option. Okay. And what about D? D goes with 2. D goes with 2. I have 2 options like that. So I have confusion between this and this option. Now cyclosporin A is basically an immunosuppressive agent which you get from Trichoderma polysporum. So B goes with 1. B goes with 1. So this could be the answer. And C, serratin, which is there, it stabilizes or lowers the blood cholesterol level. You get it from Monascus purpureus. So C goes with 4. So basically your answer for this question is going to be B. It is going to be B. Got it? This is question bank question. Fine. Fine, ma'am. Next. Which one of the following is not an advantage provided by mycorrhiza to the plants? This is an important thing. Okay. Which one is not an advantage provided by mycorrhiza to the plants? Resistance to root-borne pathogen. Does it provide resistance to root-borne pathogens? Yes. Increase in the yield quality? No. It does not increase the yield quality. I told you. Resistance, tolerance, overall increase in plant growth and development, definitely yes. And also phosphorus absorption, but not yield quality increase. It has nothing to do with that, which is why B1 is the odd one out. Okay, B is going to be the odd one out. Now what do you have? The microbial biocontrol agent for butterfly caterpillar is butterfly, all the insect group specific, the butterfly armyworm and all the things, my dear children, tobacco budworm, all of them are being treated with Bacillus thuringiensis, BT. So this is your answer. These are insects ones, okay, insect killing ones. Okay, next one, these are straight questions, right? So which one of the following is a bacterial biofertilizer? Bacterial, I'm not talking about cyanobacteria, bacterial. So answer that Anabaena, Nostoc, Acetylatoria, A and O cannot come in. What can come in? Rhizobium. Rhizobium, Azotobacter, Azospirillum, they are bacterial biofertilizers. They fix nitrogen, they do all this thing. Rhizobium is symbiotic, shows symbiotic association with the plants. Azotobacter and Azospirillum, they are free living, but they are basically providing nitrogen, they fix nitrogen. And they are all bacterial biofertilizers. Know the difference between cyanobacteria and bacteria. Cyanobacteria prepare their own food. Okay, yes. Now we enter into the next chapter, that is biotechnology, principles and processes. So we have, I think, 5 more chapters, which is why I'm increasing the speed a bit. Otherwise it can keep on going. Yes. So yes, let's go. So in the year 1963, the 2 enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. 2 enzymes responsible for restricting the growth of bacteriophage in E. coli. In E. coli, one bacteriophage was trying to grow, but the E. coli was producing some enzymes which were not allowing the growth of the bacteriophage. One of them cut the DNA and the other added the group. What? So that is being asked. So what do you think is the answer for this particular question? It's a very easy question. Listen to me very carefully, okay? So this is the thing that we have. This is the thing that we have, that is E. coli. E. coli will have its own genetic material, agreed? Now suppose a bacteriophage came in and put its genetic material inside. So the major target of this bacteriophage is to integrate this into the genetic material of the E. coli. And you have to stop that, which is why E. coli will do the first thing, that is produce restriction endonuclease, as in restriction enzymes, and that is going to cut the genetic material. But if it is producing restriction endonuclease, that can act on this particular genetic material as well. What to do with that then? You have to protect your own genetic material. So basically you will need one very important thing. You will need to add some groups over here. These groups, my dear children, are going to protect, okay, or is going to protect the E. coli's genetic material to not be degraded. Okay, so whenever methyl group is added, basically restriction endonuclease will not be cutting out there. That is why I say A is going to be your answer for this particular question. Okay, okay. Next one. Which of the following is the correct representation of primer annealing step in PCR? Primer, where does it go and attach? It attaches at the 3' end. Why? Because DNA replication happens from 5' to 3' direction. So in the opposite side, it has to be 3', then only it can do 5' to 3' thing, right? So it has to attach only at the 3' end. So here it is 5', here it is 3', there it is 3' end, but here it is 5'. So both of these are wrong. What about this? 5' again, wrong. What about this? Yes. Here at 3' and here also at 3'. So yes, probably that can be correct. So D is your answer for this particular question. Cool? Now, the chemical nature of the primer used in polymerase chain reaction is, okay, the chemical nature of the primer that you use. So usually when DNA replication happens in our body, at that time the primer is made up of RNA nucleotides out there, okay? But here it is going to be deoxyribose nucleotides. Okay, it is going to be deoxyribose nucleotides. It is not going to be ribonucleotides. It is going to be deoxyribonucleotides that you are going to use in the PCR. Always remember this. This is very, very, very important. Okay? Okay. Now, in the given diagram of PBR 322, identify the restriction site A, B, C and D. A, B, C and D. A is basically ampicillin resistant site. Okay, B is basically ampicillin resistant site. C is tetracycline resistant site. So B is ampicillin, C is tetracycline. What about A? A is the ORI site and this is the ROP site. So A is the ORI site. Yes, A is the ORI site. B is ampicillin resistant site, C is tetracycline, and D is what? HindIII. D is HindIII. For this, if at all you want to know the answer for this very, very fast, you definitely have to remember this. Again, I've taught you in master stroke. Please remember. Okay, I can make it for you guys also. Listen, here you have the origin of replication from which replication is going to begin. Here you have the ROP site. This protein is super important for replication to happen. Okay, apart from that, you have ampicillin resistant gene over here. You have tetracycline resistant gene over here. Okay, now listen. There is one restriction enzyme which you call as PVU2, that is over here. Here you have two restriction enzyme site. One is BAMH1 and one is SAL1. Here also you have two. One is PVU1 and one is PST1. Two of them. Here you have three. One is going to be EcoR1, one is going to be CLA1, and one is going to be HindIII. So listen, 1, 2 and 3. One is here, two of them are here, and three of them are there. Okay, here all of them are 1, 1, 1. Here it is 2, and here it is going to be 3 at the end. Always remember like that. In ROP, you have PVU2, here you have PVU1. Cool? Cool. Yes. Now, restriction site for EcoR1 enzyme is 5-GAA-TTC. Okay, 5-GAA-TTC3. So, 5 comes first, then comes 3. Always remember, do not go and write this is the answer. Your answer for this question is going to be A. Cool? Cool. Now, next one. Fill in the blanks by choosing the appropriate word from those given in the bracket. The plasmid DNA acts as dash to transfer the piece of the DNA attached to it. The plasmid DNA acts like a carrier. What do you call the carriers as? Vectors. What do you call them as? Vectors. So onto the plasmid DNA, if at all I'm putting my gene of interest, it is going to carry it, is going to transfer it to the next organism out there. So which is why I'll call this as a vector. So your answer for this question is this. Next question. Which of the following is not a feature of a plasmid? Extra chromosomal? Listen. In a bacteria, in a prokaryote, it has its own genetic material. This plasmid which is there is an extra chromosomal. It is going to be circular. It is going to be double-stranded, not single-stranded. Independent replication. Why? Because it has origin of replication. So this one is the odd one out. This is not the feature of a plasmid. Okay? Yes. Cool. Next one. Agrobacterium tumefaciens is used as a vector for cloning genes in the case of plants, animals, bacteria or viruses. Agrobacterium tumefaciens is for plants because this is a genetic bacteria that is there. It causes tumor in the case of plants. So if at all I remove the gene that is causing tumor and I put my gene of interest, basically it will still go and infect the plant, but this time transfers my gene of interest out there. You get it? So basically Agrobacterium tumefaciens, always remember, is there for plants. And for animals, you have retroviruses. For animals, you have retroviruses. For plants, you have Agrobacterium tumefaciens. Cool? Cool, ma'am. Next. Agrobacterium tumefaciens transform normal cells into tumor by delivering a piece of DNA known as. So listen, I'll talk about it then. So there's a plasmid. This plasmid is known as TI plasmid. TI stands for tumor-inducing plasmid. Why tumor-inducing? Because it has a segment of DNA which is known as tDNA, transfer DNA. When this transfer DNA is transferred to the plant, basically it causes tumor in them. Okay, so Agrobacterium tumefaciens transform normal cells into tumor by delivering a piece of DNA known as what? tDNA. Known as tDNA. Very, 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 very important. Please remember it. Okay? Okay. It is also known as transfer DNA. Now, which of the following is the correct order of processing of PCR? First of all, PCR means you want to replicate the DNA, right? So, DNA strand comes like this. You have to denature it. You have to separate it. That is denaturation. After denaturation, you have to attach the primers. That is known as annealing. After that, you have to extend the strand out there. That is known as extension. So, first one is denaturation. Second one is going to be annealing. Third one is going to be extension. So over here, instead of DNA, it is DNE. Denaturation. I hear you. Just a second. Just a second. So instead of, okay, I'll not, okay. Let's not have anything because I was thinking something else. Okay. So denaturation, annealing, and then extension. This is your answer. Okay, denaturation, annealing, extension. The highest temperature is required for denaturation, then comes annealing, then comes extension. Okay, so denaturation requires higher temperature, that is 95 degrees Celsius. Then you lower the temperature in annealing, then you again increase the temperature, as in it brings out to 75 degrees Celsius in the case of extension. Okay, okay. Next one. Statement 1. Sticky ends are resulted due to action of restriction enzymes. Is it? That's how you get sticky ends when you cut with restriction enzymes? Yes. Stickiness facilitates the action of DNA polymerase. Is it? Now stickiness which is there, once you get sticky ends and once they join, okay, once they join, do you think that is facilitating the action of DNA polymerase? No. Basically, who is the one that will be fixing all the gaps out there at the end? It is DNA ligase. So stickiness is helping in the functioning of DNA ligase, not DNA polymerase. So I'll say this is correct, but this statement is false. Statement 1 is correct, but statement 2 is false. Yes. Okay? Okay. So the purpose of stirred tank bioreactor is, what is the purpose of stirred tank bioreactor, my dear children? Provide anaerobic conditions? No. Aerobic bacteria you're putting, then you put anaerobic condition. No, no. Purification of the product? Addition and proper mixing of preservatives? Availability of oxygen throughout the process? Yes. Which is why you have stirrer in the middle that is going to stir things. Okay, and it has flat bedded impellers also. Got it? So your answer for this question is going to be D. Cool? Cool. Next, identify the correct statement. Hint 2 cut the DNA at specific sequence of 6 base pairs. Hint 2 was the first discovered restriction endonuclease. Okay, and it cuts the specific sequence of 6 base pairs. That is also correct. Microinjection is a technique of introducing rDNA into the plant cell. No. With the help of injection without using a vector, can you transfer the DNA? Yes, but not into a plant. Why? Because plants have a cell wall. The injection would not enter. This you do in the case of animals. This is vectorless method in the case of animals. Usually the vector that we use is retrovirus. If at all we are not using retrovirus, basically we can use a vectorless method that is known as microinjection. Over here you are not using plant cell, instead you should use animal cell. Now, biolistic is a technique of introduction of rDNA into animal cell. Biolistic is a method that is also known as gene gun, because the plant cell has a cell wall and it will need a gun. That's why you use that, and it's not for animal cell, it is for plant cell. So this is wrong. So this is gene gun method, and basically you introduce recombinant DNA into the plant cell. Over here, you use the particles of gold and tungsten and you cover it up with the DNA and then you do the bombarding. Okay? Okay. Now, retroviruses are commonly used cloning vectors in plants. Retroviruses are commonly used cloning vectors in plants? No, it's in the case of animals. So this is also wrong. So over here, the only option that is correct is going to be A. Cool? Cool. Fine. Now we enter biotechnology and its application. The 4th last chapter, and if at all you have reached until here, then write level 5, crust. Okay, let's begin. Given below are the steps in gene therapy of patient with adenosine deaminase deficiency. So they do not have the gene for adenosine deaminase, which is why we need to insert it into what? Into the lymphocytes, the white blood cells. Okay, specific white blood cells. So lymphocytes first of all have to be taken from the patient, right? So lymphocytes are collected from the blood of the patient. This is going to be the first step. Third one is the first step. So I have 2 options like that. I'll cut off these 2 options. Now I'll check my answer with this. I'll check my answer with the options. After 3, it is 4. What is 4? The genetically modified lymphocytes are infused back into the patients. No. In between that, obviously, there will be a lot of other things. You have just taken the lymphocytes. You have to genetically modify it, no, first. So, after 3, 1. 1 is what? Lymphocytes are grown in a culture outside the human body. Yes. So, they are taken first and then they are going to be cultured. Fine. After culturing them, what do? You are going to alter with the functional ADA gene. That means you are going to put that gene which is going to produce adenosine deaminase. Correct? You did the modification. And then basically last thing that you do is genetically modified lymphocytes are infused back into the patient. So that is also correct, which is why your answer for this question is going to be C. It is going to be C. Got it? Clear? Clear. Now, the next thing that we have up over here is what? The next question. The plants developed by the fusion of protoplast of two different plants are. What do you mean by protoplast? Suppose there is a plant cell and I am removing the cell wall. Over here, I am not putting the cell wall, I am just removing it. And what is left behind? Basically, if cell wall is not there, the thing that is left behind is going to be known as a protoplast. Is going to be known as a protoplast. And when I am fusing two protoplasts together, what do I get? What do I get? When I'm fusing two protoplasts together, I get hybrids, somatic hybrids. So this can be of tomato, this can be of potato, both of them fused together, I get pomato. So this is what? The somatic hybrids that I am getting. So your answer for this question is going to be B. Got it? Clear? Okay? Yes. Soma clones is a result of tissue culture. Whenever you do tissue culture and when you get product on the basis of that, that is known as soma clones. All of them would be genetically similar. Here it's not like that. Okay. Identify the incorrect statement. The transgenic cow Rosie produced milk enriched with alpha-lactalbumin. Yes, it is a human milk protein, but you're making a cow produce that by inserting the gene. That is correct. So, Bt toxin genes Cry1AC controls corn borers. We'll come to that. Human protein alpha-1 antitrypsin is used to treat emphysema. Yes, alpha-1 antitrypsin is used to treat emphysema, which is a respiratory disorder, and it is since animals cannot repeatedly produce too much copies, we make the since human beings cannot produce too much copies, we make the animals produce it. Okay, so that is also correct. The commercial production of human insulin was made possible by the transgenic E. coli. Yes, E. coli was being used for production of human insulin. That is also correct. Bt toxin gene Cry1AC controls corn borers. No. So Bt toxin, okay, Bacillus thuringiensis has a gene that is going to code for the toxin, and the toxin is very much specific. So the genes are also specific. There is Cry1AC and there is Cry2AB. Okay, and there is Cry1AB. Always remember, easy way to remember. First letter starts with C. Sorry, first word starts with C, cotton. Second word starts with B, bollworm. Since this is a bigger name, you need two genes for that. Okay, you remember like that, chumma I am saying. Okay, now Cry1AB, there is only one gene, and this is for corn borers. This produces toxins for corn borers. So only one is there, starting letter is C, and the next is B. Okay, here also it is CB only, but there it is bigger name, so you need two genes. You remember like that. Why I am telling you this is sometimes you might write 2AC over here. I do not want you to do that. First letter C, second letter B in cotton bollworms. Here it is going to be Cry1AB. Got it? That's all. So the answer for this question is going to be, okay, this is the incorrect one. Next one. Which of the following statement is correct with respect to synthesis of insulin? Synthesized as proenzyme and needs to be processed to become functional enzyme. You're talking about insulin. Is it an enzyme? It's a hormone. So this is wrong. Synthesized as prohormone and needs to be processed to become functional hormone. Yes, it is a prohormone, immature form of hormone, and it needs to be processed to become functional hormone. Synthesized as proenzyme and needs to be processed to become functional prohormone. Enzyme and hormone? Wrong, Baba. Read the options carefully. Synthesized as prohormone and needs to be processed to become functional proenzyme? Again, wrong. So you just have one answer, and that is going to be this, B. Cool? Cool. That's all. Cool. Now, crystals of Bt toxin produced by some bacteria do not kill the bacteria themselves because. So, Bt bacteria produces toxin. Why does it not kill its own self? Because it does not have the same, what do you call it, is in the inactive form inside its body. It becomes only active when you provide the ambient temperature, or basic, not temperature, the pH. Alkaline pH. Alkaline pH is found in the case of inside the insect. So once this bacteria is inside the insect, that's when the protoxin transforms into the active version of toxin. So it's basically because the toxin is inactive inside the body of the bacteria. You need alkaline pH, you get it only within the insect. Cool? Yes. Okay. Statement 1. The plants genetically identical to the original plants are known as soma clones. Is it? Genetically identical, as in I have one plant with the help of tissue culture. I took some part of it and I did tissue culture, which is why genetically I am getting same type of plants. Are they known as soma clones? The answer is yes. They are definitely known as soma clones. Now statement number 2. Soma clones can be obtained through micropropagation. Can they be obtained through micropropagation? Yes. So tissue culture is a method by which we can produce so many amount of plants, thousands of varieties of plants, which we call as micropropagation. So soma clones can be obtained through micropropagation. That is correct. So I'll say both statement 1 and 2 is correct. Okay. Somatic hybrids is different. Soma clones is a result of tissue culture or micropropagation. Always remember, they are genetically similar. Somatic hybrids are different from the parent. From the parent it was different. You got a hybrid thing, new thing out there. Now the pollen grains of a plant with 2n is equals to 48 are cultured by tissue culture to produce a callus. Listen, pollen grains of a plant. Okay, so what are you taking? You are taking pollen grains. The plant which was there, it is having diploid number of chromosomes, that is means 48 number of chromosomes. What will be the number of chromosomes in the pollen grains? Half of it only. Okay, so it will be 24 over here. Fine. Now what would be the number of chromosomes in the cells of the callus? Basically, when you are taking the pollen grains and you are growing in a medium, the cells are going to divide and going to form a tissue sort of structure. It is known as callus. You are being asked, what is the ploidy of this particular callus, or what is the number of chromosomes in the callus? It is made up of the cell, that is pollen grains. It had 24, so all of them is going to be having 24 number of chromosome itself. So your answer is going to be B. Again, read the question carefully and then only go ahead. Pollen grain has to be having haploid number of chromosome. The plant can be diploid, but here it is going to be having haploid only. Okay, so make the half of it and then everything will be the same. Next one. Statement 1. Bacillus thuringiensis produces insecticidal protein crystals called Bt toxin. Bacillus thuringiensis produces insecticidal protein. Does it produce insect killing proteins out there called Bt toxin? Yes, they do. Statement number 2. These toxins exist as prohormones before entering into the insect gut. It's protoxin, not prohormone, right? So which is why it will be wrong. So just children, sometimes they just see this, do not see this, do not do that mistake, okay? So your answer for this question is going to be statement 1 is true, but statement 2 is false. Cool? Yes, ma'am. Next question. DNA in a clone of cells followed by detection using autoradiography is called. DNA in a clone of cells followed by detection using autoradiography. So basically some DNA which is there, it is attached to some radioactive thing. And when it is attached to radioactive thing, even in between lots of other clones out there, you can still spot it. Why? Because it is attached to radioactive thing and you are viewing it under what? Autoradiography, which is why it is clearly being seen. This DNA which is attached to radioactive thingy is known as a probe. You call this as a probe. So B is your answer for this particular question. Got it? Yes, ma'am. Okay. Next thing. Use of bioresources by multinational companies and other organizations without proper authorization and compensatory payment is referred to as. So basically, suppose America, in US, my dear children, as I told you, I have not told you, okay. So basmati rice was being patented. They said that no one has the authority to produce basmati rice now, we are going to do it. But do you think that's correct? Because basmati rice origin is in India, the farmers do that, right? So without giving compensatory payment to that, can you just say that I am going to use the bioresources and I am going to put a patency on this? No, this is referred to as biopiracy. It is referred to as biopiracy. So your answer for this question is going to be A. Okay? Yes, ma'am. Fine. Next. Which of the following statement is not correct? Is not correct, okay? Most Bt toxins are insect group specific. They are definitely insect group specific. RNA takes place in all eukaryotes as well as prokaryotes organism as a method of cellular defense. RNA interference. Do you know what is that? I'll talk about that. The recombinant therapeutics do not induce unwanted immunological response in humans. Yeah, they do not. Okay, the recombinant therapeutics that we use, they should not actually induce any unwanted biological response, immunological response, right? So this is also correct. Adult onset diabetes can be controlled by taking insulin at regular time intervals. Yes, because diabetes is caused because your insulin is not being produced in the pancreas, right? So obviously, if at all you give insulin, enough amount of insulin, it's all going to be sorted. So this is correct. Now what is not correct is obviously this. But what is this? RNA interference is a method that you use for producing pest-resistant varieties of plants. So there's tobacco plant which is infected by Meloidigoin incognita, an aschelminth, or what do you call as nematode. Okay, so what this nematode usually does is that it has the DNA, it transcribes into mRNA. This mRNA translates into a protein. Now over here, I want to stop this. I want to stop the translation process, which is why what I do, it has to be single-stranded. This mRNA has to be single-stranded for undergoing translation. I am going to give this strand one more complementary strand, which is why it will become double-stranded. And do you think translation is going to happen? No. That means protein is also not going to be made. This is known as RNA interference. You're interfering over here. And this is actually a cellular defense mechanism that is found in eukaryotes, as in basically the developed organisms out there. Eukaryotes have a proper nucleus, proper cell membrane bound organelles and things like that, right? In the case of prokaryotes, they are primitive, they do not have all these things and they do not show RNA interference. Please know that. So RNA interference is something that you see in eukaryotes as a cellular defense, not in prokaryotes, but here it is given eukaryotes as well as prokaryotes, which is why this is not going to be the answer. So B is the odd one out over here. Got it? Yes, ma'am. Now, match column 1 with column 2 and select the correct options from the given codes. Column 1. Alpha-1 antitrypsin. We just studied it, but let's see. We have to read all the options first. Transposon, ELISA, retroviral vector. Okay, we know this. Now read this. AIDS, gene therapy, emphysema, mobile genetic element. Okay. Alpha-1 antitrypsin, we all know it is there for treating emphysema, respiratory disorder. So A goes with 3. A goes with 3. I have 2 options like that. Okay. B goes with 4. Transposon is a mobile genetic element. Okay. I have just keep it on side as of now. Let's go with D option first. Retroviral vector. Retroviral vector is basically used for as a carrier or a vector in the case of animals, right? So here in gene therapy I might need it. Remember, in gene therapy I need to put the gene adenosine deaminase into the lymphocytes. That is an animal cell. For that I might need it. So D goes with 2. D goes with 2. I just have one option. Basically this is going to be my answer. If D is very difficult for you, ELISA, we all know it's a diagnosis test for what? Test for AIDS. So ELISA goes with 1. C goes with 1. And you just have an option like that. So basically you got the answer. Okay, so when you have confusion, see all the options, check out for it and then only go ahead. Transposons are mobile genetic elements, as in there are genes which jumps from one place to another genes. They are known as transposons. They can also lead to very bad mutations. Cool? Cool. And now we come to organisms and population. So, my dear children, how do you feel? Is it too much for you as of now, or are you able to take it up? So I am trying my best to be in live loop and 3 hours are already over. I'm so sorry. But this is important, right? It's important, which is why I went ahead. So please listen to me. 3 more chapters are there. We will finish it up very, very fast. Okay? If at all you have crossed this level, level 6 crossed, comment in the comment section. Let's go ahead. The organisms that reproduce only once in their lifetime. Only once they're going to reproduce. Oysters, pelagic fish, salmon fish, hippocampus. Only once they're going to reproduce. You call these organisms as simulparous organisms. What do you think is the answer? The answer is salmon fish. Along with salmon, you also have bamboo. Okay, bamboo. They produce only once in their lifetime. Always remember. Birds, mammals, all of us, they will produce multiple times, but they are not like that. Okay, so salmon fish and bamboo, do not forget, do not forget, do not forget. Okay, next thing. In a pond, if there were 20 lotus last year and through reproduction 16 are added, calculate the birth rate. Okay, so previously existing one, you put it down. The newly added one, you put it up. Time period, if at all it is mentioned, you can from last year. So basically just 1 year. Okay. Now what is the calculation? You do the calculation, you will get your answer. What is your answer? It is going to be 0.8. Now, You are being asked to mention the birth rate. So you have to speak about the time. You have to talk about the organism that is producing it. So I'll say 0.8 offspring per year. Is it correct? Not correct. 0.8 offspring per lotus per year. That is going to be the birth rate. Okay, so there's a formula over here. Number of new individuals added divided by the initial population size into the time that is mentioned. So over here only one is mentioned, so it is 0.8. Okay, please mention 0.8 offsprings per lotus per year. That is very important. Okay, this is the birth rate. Now, similarly, you can be asked about the death rate. 20 were there, 10 of them died. So please know about this formula. Okay. Now, the correct order of various levels of biological organization in an ecosystem is: organisms came together to form population, population came together to form community, community came together to form ecosystem, ecosystem came together to form what? Biome. So this first option itself is the correct one. Organism, population, population, then biome, wrong. Biome, ecosystem, wrong. Biome, no. So this one is the only one. This is very easy question. I think you got it. I got it from model question paper is what I remember. Yes. Next. Jiva was growing a bacterial colony in a culture flask under ideal laboratory conditions where the resources sooner or later became limiting. Listen, this is a very, very important question. Jiva was growing a bacterial colony in a culture flask under ideal laboratory condition. Everything was ideal. But there is one thing that is clearly mentioned. Resources sooner or later become limiting. That means unlimited resources are not provided over here. Resources are limited. Which of the following equations represent the correct growth in this particular case? You are going to see logistic growth. That means there is a carrying capacity over here. There is a factor that is limiting, and that is what? Resource, which is why it cannot grow exponentially like this. There will be logistic growth. There will be a sigmoid curve that is going to be found. Okay, not a J-shaped curve, sigmoid curve. And whenever you have something like this, you say it is dN by dt is equals to Rn, intrinsic rate of natural increase, K minus N divided by K, where K is what? The carrying capacity. This capacity, carrying capacity has to be mentioned over here. If at all you are given everything is ideal, everything is ideal, it is going to be the other thing that is known as exponential growth, which goes like this. There is no carrying capacity. There the formula is dN by dt is equals to Rn. You do not include this. You get it? But here limiting factor is clearly mentioned, so please go ahead with this. So in preparatory also I saw a question which had lots of confusion. Remember, if you have not been given limiting factor, read the question carefully. Probably the answer could be exponential. Okay, this is a question that can be anything. Okay, logically speaking, exponential does not happen, but yes, if in the question it is given, you have to answer accordingly. Here it is logistic growth, okay, or S-shaped curve is being formed. S-shaped curve is being formed, okay, yes. Now, monarch butterfly is avoided by the predators because monarch butterfly is avoided by the predators, because, my dear children, it is highly distasteful. It tastes very, very bad. It's a what you call defense mechanism that this particular organism has. It is cryptically colored. No. It is highly distasteful. Yes. It produces poisonous cardiac glycoside. It actually does, but in the NCERT is not written. It is written about Calotropis, a plant, but not about monarch butterfly. So we will not also study that. Just to know, it is highly distasteful in nature. Okay. It has stinging cells on its body. No, Baba. B is your answer. Next. What does the shape of the given age pyramid A to C reflect about the growth status? Okay, so there is post-reproductive, reproductive and pre-reproductive. So the bottom one is the pre-reproductive one, okay, and then it is reproductive and then it's post-reproductive. If I say the organism in the pre-reproductive age group are more, that means in future they are going to reproduce, right? So can I say it is going to be an expanding population? Yes. Over here, if at all you see reproductive and pre-reproductive, both of them are same. So I'll say it's what? It is a stable population that I get. Then over here, I have what? The reproductive one are very less. Pre-reproductive one are very less. Reproductive one are more, right? So that means basically in future you do not have many organisms to reproduce. So can I say it is going to decline? Yes. So basically, first of all, I say this is expanding. If the lower one is more, that means it is expanding. Here it is going to be stable and here it is going to be declining. In future, there are not much organism to reproduce. Cool? Cool, ma'am. Okay. Fine. On the rocky sea coast of Scotland, the larger and competitively superior barnacle balanus dominates the intertidal area and excludes the smaller barnacle thalamus from that zone. Which kind of interaction is being depicted by this example? What do you think? So there is intertidal rock, there were the superior barnacle balanus which was there, and then there is thalamus also. Slowly what happened is that these guys excluded these guys off from this area. Balanus one, they dominated the intertidal region and they pushed the thalamus upwards. Why do you think this happened? Because of competition. Because of competition. And whenever an organism takes up the whole space, is known as competitive release. It is known as competitive release. They utilize the entire area by excluding the other organism somewhere else. Okay, this is a result of competition. Not predation, not parasitism, not commensalism. Okay, next one. Assertion. The epiphytes use the trees only for attachment and manufacture their own food by photosynthesis. So if at all you have seen the epiphytes, that is orchid growing on a mango tree. So it's just basically there for getting enough amount of sunlight and things like that, just like this, okay? It's not for it is not deriving any food from mango plant. It is not harming the mango plant. So basically, it is commensalism which is being seen over here. One plant gets a benefit, that is the orchid, which is the epiphyte, and the mango tree, it's neither getting harm nor getting benefit. So there's a +0 relationship, that is commensalism. So that is correct. That is exactly correct. Now, as reason, commensalism results in negative effects on the growth and survival of one or both of the population. Wrong. Commensalism, as I said, one gets a positive thing, and the other one is neither harmed nor affected. So it's a +0 relationship out there. So there is no negative effects that you can see. So I'll say that assertion is true and the reason is false. C is the answer. Cool? Cool, man. Next one. Abingdon tortoise in I have written the answer, but still. Abingdon tortoise in Galapagos Islands became extinct after goats were introduced on the island. Mention the type of interaction. So I have seen a picture where on the tortoise, the goats are standing. So what is happening over here is that initially you had the tortoise, but later on when goats were introduced, what exactly happened is that the goats competitively excluded the tortoise. This is known as example of competitive exclusion. It excluded the tortoise, my dear children, altogether. Okay, why? Because of the same reason. Basically, it was stronger one. The goat was a stronger one, which is why in the picture there's a tortoise. On the tortoise you have the goat. This is an example of competitive exclusion. It got extinct. Just imagine the level of competition. Both of them were having grass. Okay, anyways. Done? That's all? Yes. I think you will see more questions again in 2 marker and 3 marker from these chapters. Now we have ecosystem, the second last chapter out there. Few questions are there over here. I want to tell you one very important thing. In ecosystem, biodiversity and conservation, all of it, there is number-based question which you have to remember. I'll request you these 2 chapters, read from your NCERT very, very nicely. Each and every single line of your NCERT is important in these 2 chapters. Super, super important. Okay? And I'm not writing any levels over here. The last level is the one that I'm going to write later on. Okay. The first question that I have over here, the decomposers especially abundant in the bottom of the pond are. Decomposers especially abundant in the bottom of the pond are what? Only fungi, only bacteria, only fungi and bacteria, flagellates, fungi and bacteria. So usually we say it's FB, that is fungi and bacteria. Facebook, right? But always remember, in the case of pond, these things are changing. There are flagellates, there are fungi, and there is bacteria. Okay, please remember, it's there in your NCERT, one single line, and that came as a question. Okay, and that too in an MQP, so you can expect more of it. The correct sequence of steps in the processing of decomposition is, first thing, you have detritivores, the ones that eat on the dead and decaying matter, and you break the food down into easy ones, right? That is known as fragmentation. That is done by the earthworm and things like that. After fragmentation, whatever inorganic things are there, soluble things are there, it will go into the soil, deep into the soil, that is known as leaching. Now, we had broken down the big things into small things. Now they have to undergo decomposition, and that is known as catabolism. And while they undergo decomposition, then comes a middle stage where they are partially digested, where humus is being formed, and that is known as humification. It is known as humification. Now, this partially digested form again divides completely into inorganic things. So from organic, you're coming to inorganic things, and that is known as mineralization. So your answer for this question is FLCHM. Is there any way to remember it? FLCHM. If you have any ideas coming in your head, please share it in the comment section so that all of your friends can also get to understand it. Okay? HM, headmistress. But yes, you can think about it. Yeah? Yes. Next one. The rate of formation of new organic matter by producer is. Rate of formation of new organic matter by producer. Net primary productivity, secondary productivity, gross primary productivity. You're talking about just one producer. You're not talking about all the plants together. You will call this as primary productivity because is the primary organism out there producer? Is the primary producer only one producer is there? It's the primary producer. The productivity of it is known as primary productivity. If at all I talk about multiple plants and I say the productivity of all the plants together, then it will be known as gross primary productivity. Okay, here it's not like that, which is why it will be just primary productivity. Remember. Okay, yes. Now, which of the following food chains is the major conduit for energy flow in terrestrial and aquatic ecosystem, respectively? So in terrestrial, which one is the major conduit of energy? Grazing food chain, detritus food chain, or what? It is actually detritus food chain, which is the major conduit of energy over here. In aquatic, it is going to be grazing. Why? Why like that? We expect the opposite to happen. We expect the opposite to happen, right? But why is it like that? Listen, in detritus food chain, decomposers plays a major role. Decomposers cannot undergo decomposition. Okay, cannot help in decomposition if at all there is no moist environment. In the aquatic water bodies, the temperature is very cool, which is why it cannot favor detritus food chain. Get it? Why? That's the reason it has grazing food chain in there. In terrestrial, on the other hand, there will be detritus because you have the moist environment, aerobic nature, all the things that you need. Okay? So please remember this offbeat question. Okay, so here I have marked wrong. The answer is this. Okay? Okay. Next one. If 10 joules of energy is available at the producer level, then the amount of energy present at the level of secondary consumer is. Okay. 10 joules of energy is available at the producer level. So suppose plant is there. Plant is eaten by a rabbit. A rabbit is eaten by a tiger, right? So this is going to be your producer. This is going to be your primary consumer. And this is going to be the secondary consumer. Right. So over here, I am saying 10 joules of energy is right up over here at the producer level. The amount of energy present at the level of secondary consumer, this level. So 10% of 10 is 1 joule. 10% of 1 is going to be 0.1 joules. So your answer for this question is going to be 0.1 joules. You get it? You have to know about the secondary consumer, primary consumer and all of it. Cool? Yes. Going ahead, what do we have? Next thing, question number 6. Given below is a table of factors affecting microbial decomposition. Okay, and plus means favoring and minus means inhibiting or lowering. Select the correct option. So you have to say which one favors microbial decomposition. Decomposition process is going to be helped with the help of the ones mentioned over here. That is denoted by plus. Okay, let's go. Then we'll understand. High temperature. Do you think it favors decomposition? Here it is written plus, here it is minus minus. I think it is this high temperature actually favors decomposition. Lack of oxygen, does it favor decomposition? It does not. It does not. So this one is the answer. It does not. There should be oxygen. Moist environment, does it favor? Yes, it does. Lignin and chitin and detritus, in the detritus, it does not favor. Lignin and chitin is difficult to decompose, which is why it does not favor. So which is why your answer for this question is going to be B. High temperature you need, lack of oxygen is negative, is going to have a negative impact. Moist temperature is going to have a positive impact. Lignin and chitin are going to have a negative impact. On the other hand, if the detritus is rich in carbon as well as nitrogen, it would have a better impact. Okay? Okay, just a second. Okay, just a second. Here, this one is the answer. This is not. Okay, I'll quickly change. I think that's better. Just a second, okay? Okay. Yes, so I have marked it. I didn't get the color. Just I marked in red. I hope that works, yeah? You will be able to see in the PDF. Now, what type of ecological pyramid would be obtained by the following data? Okay, my voice is going. Secondary consumer 120 gram, primary consumer 60 gram, and primary producer 10 gram. So over here, producer is at 10 gram, primary consumer is at 60 gram, and secondary consumer is at 120 gram. Gram as in you're talking about biomass. And this is, do you think upright pyramid of biomass? No, this is inverted. This is ulta pyramid of biomass, right? Top one has the highest biomass. This is usually seen in the case of oceans. In the case of oceans, my dear children, the producer is phytoplankton. They have very less biomass. And then it is eaten by zooplankton, they have higher biomass. The small fishes have even more higher biomass. So this is inverted pyramid of biomass, my dear children, and this is usually seen in the case of oceans or aquatic things. Okay? Yes. Next question. Assertion. Some aquatic ecosystem has inverted biomass pyramids. We just studied about it right now. Yes, it does. Pyramid of energy is always inverted in such, is also inverted in this case. Energy pyramid can never be inverted. It will be always upright. Energy is going to go less. It cannot be more at the top and can be less at the bottom. No, that is not true at all. So assertion is true, but the reason is false. Assertion is true, but the reason is false. C is your answer. Cool? Cool. Next, the annual net primary productivity of the whole biosphere is estimated approximately. So both of these questions are very related, which is why I put it up. This is there in your question bank. Annual net primary productivity of the whole biosphere. What do you mean by net primary productivity? Listen, gross primary productivity means all the plants producing their things. That is the productivity of all the plants come together. That is GPP, gross primary productivity. Now, obviously, it is not available completely to the next organism. Some of it is lost as respiratory losses, as in some of it will utilize this for itself also. That is why that is what I call as respiratory losses. So GPP minus R is equals to NPP, net primary productivity, which is actually available for the next group of organism out there. Okay, over here the annual net primary productivity of the whole biosphere is estimated to be 170 billion tons. This is the actual answer. Now out of that, the annual net primary productivity of ocean is only half of it, and that is basically 55 billion tons. Not even half, it's more, it's lesser than that also. So it will be 55 billion tons. Just a second. Okay, so this thing is very, very important, and with this we complete the other chapter as well. And just give me a second. Okay, sorry dear children, someone came. Okay, so I was completing this. Please remember this is a very important question. Oceans contribute less. Okay, the terrestrial area is contributing more than oceans over here. So 170 billion, not million, please remember. Here also 55 billion is there, not million. Cool? Cool. Next thing. Biodiversity and conservation. This is the next chapter that we are having, and with this we end the whole MCQ portion. And I think, my dear children, this video had helped you in some ways, and if at all you have reached up to this particular level, put level 7 crossed, and it goes like this. The IUCN Red List documented the extinction of species in the last 500 years. Include how many species? This type of questions are coming from this chapter. So you have to remember the numbers. It is 784. It is 784 species extincted in the last 500 years. Please write this particular number. And out of that, 338 were vertebrates, 359 were invertebrates, and 87 of them were plants. You can get a next question related to this also. 338 vertebrates, 359 invertebrates, the more number of invertebrates got extincted, and 87 plants. This is the data that you need to remember. Okay, let's try it out once. 338 vertebrates, 359 invertebrates, 87 plants. Okay, total number is 784. 784. They can give 847. Listen, you see the options. You get to understand things, right? Yeah, next thing. The following are the in situ conservation approaches except. In situ means conserving in their natural habitat. So following are the in situ conservation approaches except. National parks, it is in situ or ex situ? What do you think? In the natural habitat. Yes, it is in situ. Wildlife sanctuary, yes, it is in situ. Biosphere reserves, yes, it is in situ. What about botanical gardens? It is not. You take the plants, you grow it somewhere else in some areas, right? This is botanical gardens. This is ex situ conservation method, not in situ, which is why this one is the odd one out. Got it? Yes, ma'am. Fine. Next. Alexander van Humboldt observed that within a region, species dash increased with increasing explored area but up to a certain limit. So basically he said that when you move on to a different area, if you keep on going on, what is going to happen? The species richness is going to increase. There are more number of species that you're going to see when you explore more area, but this happens only up to a certain limit. And he gave a formula, he gave a graph and everything for that. We will be discussing about that also over here. So here it is species richness that you have to write. Okay, the term that you have to write is species richness, not diversity. You write species richness. Alexander ends with R, so you can remember richness like that. Cool? Yes. Honestly speaking, I'm tired. My legs are shivering now. Okay. The genetic diversity shown by Rauwolfia vomitoria growing in Himalayan ranges produces an active chemical called what? A genetic diversity shown by Rauwolfia vomitoria. So there is a species, or basically there is a plant, Rauwolfia vomitoria. Okay. Now, genetically there are different strains of the active chemical that is being produced. And which is why we say that it is an example of genetic diversity that is being shown over here. What is the component that is being produced? It is reserpine. It is what, my dear children? Reserpine. This is very important. It is reserpine. On the basis of how much amount of reserpine is being produced, they are being genetically diversified. Okay, if you have got the confusion, I'll just explain again. Genetic diversity shown by Rauwolfia vomitoria growing in Himalayan ranges produces an active chemical that is known as reserpine. Depending on how much quantity it produces, we say that is genetically diverse in nature. Okay? Yes. Okay. So as per the species inventory, 70% of the species recorded are, as in the most number of species which are there, they are recorded are plants, fungi, bryophytes or animals. What do you think is the answer for this? The largest number of species recorded are animals. They are definitely going to be animals. D is your answer for this particular question. Cool. Very important thing. Okay. Now, in the animals, if at all you are asked, most of them are going to be insects. Insects are the largest group in the animals. Now, always remember, in the case of plants, okay, in the case of plants, they have added fungi also. And they are saying fungi is the one that is having the highest. So, in overall, it's the animals. In the animals, it is insects. If at all you're talking about plants, they have added fungi group and they are saying that fungi is the highest. Okay, fungi is the highest, and yeah, so please remember that. And yes. Now, in general, species diversity dash as we move away from the equator towards the poles. When we move away from the equator towards the poles, what is going to happen? The species diversity is going to decrease. Why? Because poles is not a better environment, does not provide a better environment to survive, for the survival of the organisms. There is frequent glaciation, things like that, which is why the species diversity is going to go less, not high. So I'll say it decreases. It is going to decrease. Cool? Next, the graph given below shows species area relationship. I told you this is Alexander von Humboldt gas. So, which of the following equation currently represent the curve? So, over here you have the area and over here you have the species richness. Listen, Alexander von Humboldt said that yes, it is going to keep on growing but only up to a certain limit. So, basically I can not say that S is directly proportional to A. Species richness is actually not directly proportional to the area explored. Okay. It's somewhat it is not. So how do you express that then? You basically remove the proportionality quotient, you put C over here and you put A, and then you say up to a certain limit that is represented by Z. So S is equals to CAZ. This particular line is representing S is equals to CA to the power Z, this much amount. Okay, then species area, and this is related to this much species richness, and area is related to in this much particular amount. Now, if at all you take the log value of this, then it will be log S is equals to log C plus log A. And what do you do with the Z? Z you put it over here. So log S is equals to log C plus log A. Z, which was an exponent over here, you put it over here. And this is represented by a line like this. This is known as the slope of regression. Z represents over here the slope of regression. It means that it shows the relationship. So suppose over here I say that this much is the area. This much is the area that I'm exploring. Okay, or basically let me make two lines. This is one line, this is another line. So here, with this much area explored, the species richness is what? Less. Species richness is what? Less. Now if at all the slope is like this, I can say with this much area explored, the species area richness is this. This slope is usually found in larger areas. So even with less area, you can find more species richness. This is found in larger areas. Mostly, if at all we see this type of slope of regression. Where the area explored is more, the species richness is not that great. So over here, I'll say the Z value is going to be less. This is happening in the smaller areas. This happens in the larger areas. In the larger areas, it goes up to 0.6 from, it goes up to 1.2 is what I remember. Just a second. Okay, I'll just check it. 1.2 was the thing. Yes, it was 1.2. So where was I? Yes, here. Are you? Yes. 0.6 to 1.2. This will be the condition when the area is large. This is not the usual one. The usual one has a Z value from 0.1 to 0.2. From 0.1 to 0.2. This is very, very important. Most of the ecologists say that the Z value is from 0.1 to 0.2. Only when the area explored is very large, at that particular time, the Z value goes up from 0.6 to 0.6 to 1.2. Very, very important for you to know. Got it? Here, which of the following equation currently represents the curve? This curve is represented by S is equals to CAZ. So A is the answer for this particular question. Cool? Now, the given pie diagram represents the proportionate number of species of major taxa of vertebrates. Identify the group A and B. So A is this, B is this. You have to identify. So A is basically fishes. And here you have to just go like farm. It's very easy. F-A-R. This is going to be birds, and my dear children, this is going to be mammals. So in the farm, you have to include B also, that is birds. So what is A over here? It is fishes, and what is B over here? It is already birds. Okay, so A is fishes and B is birds. Your answer for this question is going to be B. Okay. So farm, you write, but in between that you include the birds. So fishes is the one that occupies the most in this, in the case of vertebrates. Now, select the incorrectly matched pair. Tannins are narrowly utilitarian. What is tannin, my dear children? Tannin is a product that you get from the plants, okay? So, when you get products, whenever you are safeguarding the nature, ecosystem, because of the direct benefits of the products which are there or ecological services which are there, you say you are saving them under narrowly utilitarian point of view, okay? Not so ecological services, I'll say the products, directly products. If at all I'm gaining some products from ecosystem, and which is why I'm safeguarding the nature, it is safeguarding under narrowly utilitarian point of view. So this is correct. Tannins, resin, gums, everything I'm getting from the nature, and basically for that I am safeguarding the nature. Now, endemism means species confined to a particular region and not found anywhere else. Is it that? Yes. And these species are known as endemic species. That is also correct. Lungs of the plant is Amazon rainforest. Is it? Yes. Rajasthan is sacred groves. So in Rajasthan, sacred groves are the last refuges for a large number of rare and threatened plants. In Rajasthan, you have Aravalli Hills, that is a sacred grove, definitely, but it is not the last refuge for a large number of rare and threatened species. Last way to conserve the nature is sacred groves in Rajasthan. Do you believe it? It's not. It's actually Meghalaya. In Meghalaya, sacred groves is the last refuge. Always remember. And Meghalaya, you have Khasi and Chansha Hills. They are the sacred groves. Cool. So this one is the odd one out. Next one. All of the following are included in ex-situ conservation except wildlife safari parks, sacred groves, botanical gardens, seed banks. Which one is included in ex-situ except? So wildlife safari parks, is it ex-situ? Yes. Wildlife national park is different, wildlife safari park is different, okay? Or wildlife sanctuary is different, sorry. Wildlife sanctuary is in situ, wildlife safari park is ex-situ. Sacred groves, my dear children, it is in situ. Why in situ? Because we are conserving organisms in their natural habitat. That is why you call them in situ form of conservation. Basically, I have some religious attachment to a place and I am conserving the nature, but accidentally the organisms who are there, they are also getting conserved. So this is in situ conservation. Botanical gardens ex situ, seed banks also ex situ. So this one is the odd one out because this one is in situ conservation and all of the others are ex situ conservation. Okay. Assertion. The variation in Rauwolfia growing in the different Himalayan ranges might be in terms of potency and concentration of the active chemical, that is rhizopine, that the plant produces. Yes. So there is no difference apart from that. It is just that there are different types of Rauwolfia and we differentiate it on the basis of the different potency and the concentration of the chemical that they are producing, which is known as rhizopine. Correct. Reason. A single species might show high diversity in the genetic level over its distributional range. Yes. In the distributional range, a single species is there, but they are showing different variations, genetic variations, because it is happening in the genetic level, not in the species level. It is happening in the genetic level, which is why different potencies or different concentration of chemicals are being produced over there. So this is correct. In the genetic level, they show variation, not in the species level. Very, very important. So I'll say both assertion and reason is true, and the reason is the correct explanation of assertion. Okay. Next. Assertion. Speciation is a function of time and tropical regions has got a long evolutionary time for species diversification as compared to temperate regions. Do you think so? Has tropical regions got more time than temperate regions? Yes. In temperate regions, there is more frequent glaciation and things like that happening, not providing enough amount of time, which is why I'll say this is correct. Reason. Temperate regions have undergone frequent glaciations in the past, whereas tropical regions have remained relatively undisturbed for millions of years. Yes. Okay, temperate regions have underwent so much frequent glaciation, which is why you cannot allow the organisms to stay up there. Organisms which are there, my dear children, they die because there is frequent changes in the environment up there. But in tropical, it is not like that. So again, the assertion and reason is true, and the reason is the correct explanation of assertion. Okay, next one. Robert May places global species diversity at about. What is the global species diversity, my dear children? It is 7 million. I added this question from the question bank because I thought these type of questions are important for you guys to practice. Robert May places global species diversity at about 7 million, not billion, 7 million. There it was 170 billion tons. That is net primary productivity of the whole thing, right? Here it is 7 million, not billion. Please know, this is Robert May, global species diversity. Next, the share of global species diversity of India is, the share of global species diversity of India is what? 8.1, 8.5, 9%, 2.4%. It is 8.1%. It is going to be 8.1%. Again, in your NCERT, and so many other terms are also given in your NCERT, which is why I am requesting you read it. Okay, next one. According to ecologists, the value of Z lies in the range of, as I told you, the smaller region, it is 0.1 to 0.2. In larger areas, it is 0.6 to 1.2. But you have to specifically focus on, most of the ecologists say it is 0.1 to 0.2. Okay, only in the larger areas it is like this. So which is why your answer can be picked up. This is a thing that most ecologists say. That is why this one. In frugivorous, in areas where you have frugivorous plants and animals, my dear children, there the Z value goes up to 1.15. Okay, 1.15. In the places where you have frugivorous, animals and other things, Z value goes up to 1.15. Remember that also. Okay. So this is in the larger area, this is generally in the smaller area, but this is what most ecologists say. Fine. Next thing. The Earth Summit was held in Rio de Janeiro in the year. Earth Summit was the first thing to be held, then came the World Summit. So Earth Summit was held in Rio de Janeiro, do not forget that, and it happened in 1992. This one happened in 2002, both of them 2-2. Okay, this was World Summit and this was on sustainable development, okay, and it was held in Johannesburg. So first one in Rio de Janeiro in 1992, Earth Summit, then World Summit in the year 2002 in Johannesburg. Very, very important. Cool? Done. And I think that's how, yeah. There are a few more things in terms of numbers that you need to know. 34 biodiversity hotspots are there, not 32. In a question bank question, I saw 32. It's wrong. 34 biodiversity hotspots, and these hotspots are rich in endemic organisms, the organisms that you're not finding anywhere else. 3 of these hotspots are Western Ghats in India, okay, Western Ghats and Indo-Burma and Himalayas. Just a second. So Sri Lanka has also been mentioned over here, just a second, sorry. So Western Ghats and Indo-Burma region and Himalayan region of India are the hotspots. Not know why Sri Lanka has been mentioned over here. And my dear children, although all biodiversity hotspots put together cover less than 2% of the Earth's land area, only 2%. But the number of species they collectively harbor is extremely high, and strict protection of these hotspots could reduce the ongoing mass extinction by about 30%. By how much percentage can it reduce? 30%. Trust me, these type of questions have come. Now, India now has 14 biosphere reserves, 14 biosphere reserves. This is different from biodiversity hotspot, okay? 34 biodiversity hotspot, 14 biosphere reserves, 90 national parks, 448 wildlife sanctuaries. This is in India. This is all over the world. 34 biodiversity hotspots all over the world, 3 of them in India, Western Ghats, Indo-Burma region, and also the Himalayas, okay? And over here, 14 biosphere reserves, 90 national parks and 448 wildlife sanctuaries out there. Okay, and if at all we, even though the biodiversity hotspots just occupy 2%, but they can reduce the mass extinction by 30%. I know their number, I know it's like ratification, but yes, in this chapter you have to do this. Sad truth, but yes. And we end up this, if at all you have crossed this level. I know the level is different. Level 10 crossed you, right? Okay, level 10 crossed, because this is like level 10, right? So my dear children, wishing you all the best. I hope this video had helped you. Definitely read the NCERT from your end. Wishing you all the best, and if you have any queries, do let me know. And lots of love to all of you. Please take care of yourself, and you are doing great. Okay, bye bye. Lots of love.